femme fatale. Mais je ne buvais pas, je ne me droguais pas, je n'avais aucun complexe. Je suis beaucoup trop normale, ça me vexe. Je ne suis pas parisienne, ça me gêne, ça me gêne. Je ne suis pas dans le vent, c'est navrant. Aucune bizarrerie, ça m'ennuie, ça m'ennuie. Pas la moindre affectation, je ne suis pas dans le ton. Je ne suis pas végétarienne, ça me gêne, ça me gêne. Je ne suis pas karatéka, ça me met dans l'embarras. Je ne suis pas cinéphile, c'est débile, c'est débile. Je ne suis pas MLF, je sens qu'on m'en fait grief. M'en fait grief. Bientôt, j'ai fait connaissance d'un groupe d'amis vivant en communauté dans le même lit. Comme je ne buvais pas, je ne me droguais pas, je n'avais aucun complexe. Je crois qu'ils en sont restés tout perplexes. Je ne suis pas nymphomane, on me blâme. Je ne suis pas travesti, ça me nuit, ça me nuit. Je ne suis pas masochiste, ça existe, ça existe. Pour réussir mon destin, je vais voir le médecin. Je ne suis pas schizophrène, ça me gêne, ça me gêne. Je ne suis pas hystérique, ça se complique. Oh, dit le psychanalyste que c'est triste, que c'est triste. Je lui dis, je désespère, je n'ai pas de goût pervers. De goût pervers. Mais si, me dit le docteur, se rhabillant.
This is on. Hello? Yeah. It's working. It's working. Oh je, was macht denn der da mit der Kamera? So, hello everybody. We will start now our meeting here in Prague. Warmest welcome to all of you, to all participants in person, and of course to all participants who join us now online. They see us, but we won't see them. I hope we were successful in putting together an interesting program so that for every one of you you find something, everyone should find something for himself. I don't want to talk too much. I want to have some organizational remarks. We have tomorrow evening our dinner cruise, our gala dinner on the Vltava River, and there are still some places free. So if somebody is not recorded and wants to join us on the desk, he can buy a ticket. That's the one thing. The other one thing, which is very important for us, our website, our ESFH website is under construction, so it's not possible to become a member online. So you will find tomorrow on the desk form where you can write your name and your email address and your profession and your affiliation. You will also find my email address, and if somebody wants to become a member, please send the request to me, and I will organize everything. At the end of the year, at the end of summer, our website should be working, and then everything is online possible. But at the moment, we have a workaround progress. Thank you very much. Yes, I hope we have now all together a scientific, an educational, and very important, also a very nice meeting. And now I want to hand over to my co-organizers, Stenka Kasova and Volker Witt. Who is the next? You? Okay.
Ladies and gentlemen, it's a great honor and pleasure for me to welcome you in the ESFH conference in Prague. But this is not the first ESFH conference held in Prague. The previous one was organized in 2003. It is 23 years, a very long period, and many things in apheresis technologies evolved significantly with new therapeutic possibilities emerging. But one thing remains unchanged. It is the joy of meeting with colleagues, with an opportunity to share stimulating discussions. So I would like to wish you a very successful conference with many new ideas, many new insights. And I also would like to wish you a pleasant stay in Prague in these early spring days. Thank you.
We organized the perfect weather.
Yeah, thank you very much. And a warm welcome also from me as I'm from the AAA, Austrian Apheresis Association. It's a good name, isn't it? But it's the first time that we have co-organized with the European Society for Hemapheresis, and it's really a pleasure to find you all here. I think we are all friends. Most of you I saw sometimes in the history, and I hope that we have nice discussions with this wonderful program in the next 2 days. And I want to say, please feel free and ask and discuss, because that is one of the most important things, that we go into discussion, that we have a fruitful discussion about where to go. And I think in these times where so far roughly 17,000 children died in the last 3 weeks, I think it's our purpose to make something more better and to hope that this ends now. And I think for that we are responsible for our medicine, and we should go ahead to make a good apheresis. Even there is a big competition with the pharmaceutical industry, with small molecules, and I think we should discuss our therapies in this context and bring it a little bit more forward. Thank you.
And now it's a pleasure to me to invite Michele Lozano for his talk. He is the winner of the Helmut Borberg Award. And Andreas Buser, because he is moderating this session.
Slides. Can I have the first slides, please? No, it's not. It's the other one, the one with M. Yes, thank you very much. So, hi, my name is Andy. I am from Switzerland, and it is a huge honor for me to be here and to present the winner of this year's Helmut Borberg Lecture and Award. For those of you who might not know who Helmut Borberg is or was, he was a specialist for internal medicine and a pioneer in apheresis, and he was in 1984 one of the founding members of the ESFH, and he was an active member of the ESFH board from the beginning. Eventually he became Congress president in 87, and he was president of the ESFH in 87 until 89, and then he took the very important role of treasurer of the ESFH, and he has founded a firm basis for the finances of the Congress of our society. After his retirement, he didn't retire, but he was also busy in a private hemapheresis center, and he was looking after reopheresis, which is very interesting. So now you know who Helmut Borberg was. And this brings us to the laureate of today, to Miguel Lozano from Barcelona, from Catalonia. He is the chief of the chemotherapy section at the Department of Chemotherapy and Hemostasis at the Hospital Clinic in Barcelona. He is a specialist in hematology and chemotherapy, and he is one of the big specialists today in the field of apheresis, and this is highlighted in his many roles that he has and will have in the future. For instance, you see here, he's the president-elect of the World Apheresis Association, where he was a vice president for the past 6 years. And he was a long-standing president of the ESFH, and I had the pleasure to work with him in the board at this time. And he was also the president of the Spanish group of apheresis. Well, but that's not nearly enough. As you know, and you are avid readers of all these wonderful journals and books you might have read, he is for sure, he was in the editorial board, or he might even be the editor of this. For instance, he is since 2020 the chief editor of Vox Sanguinis. He was an associate editor of Transfusion. He is in various editorial boards for many other different journals. And he was the chair of the AABB press editorial board, so you might have read all these books about the principles of apheresis, or you might have read the books of preparing platelet and blood components. That was all done by Miguel and under his watch. And he was also a member of the BEST collaborative, which is advancing also transfusion and apheresis. Of course, he has been publishing, like, I mean, you know, the funny thing was always, when I was discussing with Miguel, I said, oh, wouldn't it be interesting to study, for instance, B cell and T cells in consecutive photopheresis patients? Oh yes, we published that 2 years ago. Okay, thank you very much. So, I mean, everything I always wanted to do, he has already published. So that was the excuse of me being a very lousy scientist as compared to Miguel. You see here his oeuvre, and then of course his research foci, you know, therapeutic apheresis and cellular therapy apheresis, several therapies for the sake of it, and pathogen inactivation of blood components. That's how I stumbled upon Miguel Lozano 12, 15 years ago or so. Yeah. I mean, in my case, 25 kilograms ago. And of course, he's also very active in bone marrow and stem cell transplantation. He's not only a brilliant scientist and a mentor in apheresis, he's also a very, very fantastic human being, a funny man who with a beautiful sense of humor. And you see this also in his activities around the work and around conferences, always immaculately dressed, perfectly dressed, even with a sombrero as he has there. And so this makes him this year's perfect laureate of the Helmut Borberg Award and Lecture. And the title of his lecture is The Past, Present and Future of Apheresis. Professor Lozano, thank you very much for everything you have done. The floor is yours.
Thank you very much, Andy, for this kind introduction. And thank you very much also, ESFH, for giving me the honor of giving this year the lecture for recognizing Professor Wolver activities inside the society. Thank you very much. Well, the topic of my presentation is to discuss with you the past, the present, and the future of apheresis. Well, let's start by the past. Well, to speak about the past of the apheresis, we have to go almost 150 years ago, and to speak about one Swedish engineer. And I would like to mention here that the importance of the engineers that have had in the development of the apheresis activities. And well, this man patented something for the first time that was rotating for separating something. In this case, obviously, the rotation was manual, and what was inside here was the milk, because the machine was to separate the cream from the milk. And it was funny because it was, you can see that it was patented at the end of the 19th century, but the machine was still working, but obviously electrically, so it's still in the market. And I would like to draw your attention to this specific part that was rotating for separating the milk, because probably you would recognize this in a machine that was patented almost in the 50s, by this man, Edwin Kohn. He was a biochemist interested in the separation of the proteins. And at that time, after the Second World War, the Americans were interested in having plasma because they were thinking that with the potential threat of a nuclear war, a lot of plasma derivatives would be needed. So they asked to the Dr. Kohn, at that time working at Harvard, to develop a program to produce plasma and to separate the plasma into components, immunoglobulins, albumin, and so on. And so he developed a machine to separate the blood. The red blood cells were here, and the plasma was in the middle. But this idea was in collaboration with other industries. And one of them was a small company at that time that was called Emonetics, that patented something similar to the machine that was developed by Dr. Kohn, but obviously introducing new things. For instance, it was plastic, and it was of a single use. And that was the origin of the bowl of Latham. By the way, let me remind you that we have talked about Professor Kohn and Côte de Laval, and this is the name of the lecture award of the World Apheresis Association, just to recognize the role of those two persons in the world of the apheresis. Obviously, Emonetics was improving the design, the functioning of the machine, and for instance, this is a picture of the 70s of a V50, at that time mainly used for collecting plasma and platelets. Again, we have to talk of another engineer. In this case, George Judson, IBM engineer. He was unlucky because his eldest son developed a chronic myelogenous leukemia. At that time, in the late 50s, early 60s, the only treatment that we had for chronic myelogenous leukemia was just drawing blood, centrifuging the back, removing the supernatant, the leukocyte, and giving back the red blood cells to the patient. That was the treatment that George Judson's son was receiving in Washington, in the National Institute of Health. But there was also a doctor, Dr. Frederick, that was interested in separating blood in a more efficient way. And Dr. Judson suggested that obviously that particular treatment done manually could be done automatically. So he suggested to develop a machine to perform that particular treatment. So he, unexpectedly, he convinced the responsibles of IBM to allow him to develop that particular project. So he was allowed to stay at the National Institute of Health in Washington for 1 year. So he started almost from scratch. And this is a prototype of the initial machine. And in 1.5 years, he was able to produce the first separator, IBM 1920. That was in 1965. Obviously, the machine could separate leukocytes, but at that time, the major use was for collecting platelets, as you can see here. But that machine was just mechanical, just plastic. You see here the screws, and after each usage, the machine had to be dismantled, cleaned, and sterilized. So imagine at that time the work that they represented. Something happened in the 70s because some engineers again developed a system that allowed to have a continuous flow machine. It was clever design. Just it was an adaptation, making the centrifuge with a separated holder that made that with each turn of the centrifuge inside, 2 turns, unwind the twist produced by the rotation. This allowed to have just a continuous tubing from the patient to the machine and back to the patient. This allowed the introduction of the first automatics as apheresis separators. In the late 70s, for instance, at that time, Baxter Fenwal introduced the CS 3000. Here you can see the system where they're rotating inside twice with respect to the external part. Obviously, IBM identified that the apheresis system was not their interest in the business. So they sold everything related with apheresis to a company named Cope Laboratories in the US. They continued working, and obviously they introduced more technology, and also they introduced for the first time the software to control the machine. So we had in the market in the late 80s the Cope Spectra. We have talked a lot about what was happening in US. But you know, also things were happening here in Europe. And for instance, there was a company, Italian company called Dideco, that was producing also a apheresis machine. This is the Dideco Vivacell, the model introduced in the early 80s. You can see here, and the use. Imagine all the controls that the machine had and how difficult it was for the operator to control the machine. Unfortunately, about 20 years ago, Dideco stopped being in the business of the apheresis. But also in Germany. In Germany, a company called Fresenius, probably you have heard of it, was starting developing apheresis machines. The first one, the model, it was, this is the second model actually, the AS Tech 2004, and that was updated with the model Comtech model in 2001. Fresenius stopped developing Comtech machine because they bought Fenwal, the Amicus, and so they now are selling the Amicus machine in some countries, while they are keeping also Comtech in some others. But with the time, probably you are familiar now with those models that were introduced in the 2000. You have heard, you know, the Spectra Optia, the Monetics, and the Amicus. This is the present, the machines that we have nowadays in our apheresis units. What was the past clinical practice? What people did with those machines that we have been reviewing? I found this publication performed by the colleagues that were working at the apheresis unit of the hospital clinic in Barcelona. In 1985, they published they performed an analysis of the adverse effects of plasma exchange. They analyzed 748 plasma exchanges performed in 75 patients. In those patients, they performed 651 procedures performed with the IBM 2997. It was the second model of the IBM. And also they use the discontinuous cell separator, the Emonetics H30. These are the diseases that they treated. Probably you will recognize that most of them are the same diseases that we are treating nowadays, mainly autoimmune diseases. But for me, it's really striking that just in that time, they treated just one patient with TTP. You know why? Because probably at that time, TTP was not a well-known disease entity, so it was not diagnosed. Or when it was diagnosed, it was too late for the patient. For me, what is striking to see the side effects that they found. The total incidence, they reported 18% of side effects. But look at here, 3 patients developed sepsis during treatment with repeated and immunosuppressive drugs. All of those patients developed hypogammaglobulinemia. And can you imagine the combination of patients receiving immunosuppressants for treating the autoimmune diseases combined with hypogammaglobulinemia generated by the plasma exchange? The final result was that the patient got infections and some of them developed sepsis and 2 of them died in the end. And also amazing, all of patients that received fresh frozen plasma as a replacement solution developed non-A, non-B hepatitis in the 80s. Shocking news now, because now we know that non-A, non-B is hepatitis C. We have identified the virus, we can perform a screening, and we can inactivate also the plasma. But you know, always we have been talking about therapeutic apheresis, but you know, most of the apheresis activity is done in donations because we are collecting plasma and platelets by apheresis. And here they also reported the experience in 1986 of platelet apheresis. They reported 718 platelet apheresis, and they were using all the machines that at that time was used: Haemonetics 30, Fenwal CS 3000, IBM, 2 different models, another Haemonetics, the next model of the Haemonetics, and it's surprising because they reported that with Haemonetics 30 and IBM 1997, they collect huge amount of platelets. You can see 5.4, 5.9 times to the 11. But unfortunately, look at the contamination of leukocytes. Very huge. Obviously, next models, next technology like CS3000 or double stage IBM or Haemonetics, obviously the platelet yield was lower, but fortunately also with a lower leukocyte content. And you know, the more leukocyte, the more side effects with the transfusion of those platelets. This is just to show you the different machines used. I would say certain percentage of reactions during the procedures, and that the machines with some of them with high percentage of paresthesias, that fortunately nowadays with the modern apheresis separators that we have, this is much lower. Well, what is the activity nowadays in the world of the apheresis? Let me show with you a survey that we performed in Spain in 2011 about the activities of in Spain. It was done by the Spanish apheresis group. In Spain, we have the country divided into 17 autonomous regions, different autonomous regions that listed here. We sent surveys to all of them, and we receive a nice percentage of response of the different centers. Most of them performing apheresis, and most of them, they were blood centers, blood transfusion centers. What about therapeutic apheresis? Again, we receive a nice response rate, and with most of the hospitals, because mostly those were hospitals, they were performing therapeutic apheresis in the hematology department, because this survey was sent to the members of the Spanish apheresis group, mainly hematologists working at hospitals, but some of them reported that in some of those centers, therapeutic apheresis was performed in another department, mostly nephrology. I have already mentioned that donation is the main activity for apheresis, and you can see here that at that year in Spain, we collect data of more than 44,000 apheresis donations, and most of them were multi-component, mainly single platelet apheresis combined with plasmapheresis collection. Also plasmapheresis, single platelet apheresis, and so on. Just residual numbers for red blood cell collection. What about therapeutic apheresis in adults? In adults, the most performed procedure was plasma exchange, but with 2,000. Remember that we were talking that with apheresis donations, we collected data of 44,000, and with for therapeutic, just 6,000 procedures. Mostly plasma exchange, stem cell collections, erythrocytopheresis, and at that time, remember that we were in 2011, extracorporeal photopheresis, but this percentage nowadays is much higher. Also LDL apheresis and so on. What about pediatric patients? Because we also collect data of some hospitals that were treating pediatric patients. Well, you can see here, most of the treatment were performing child is extracorporeal photopheresis, because those centers are very active in stem cell transplantation. And so they are treating GVHD with ECP, but also as a second one, plasma exchange, stem cell collection, LDL, and so on. We have been talking about Spain. What about Europe? Well, there is no data. So we had to go to use a creative measure. So we asked the three main manufacturers of disposable for plasma exchange in Europe, Fresenius, Terumo, and Haemonetics, and we received data of the disposables units sold in different countries. And for us, it was a surprise, because you can see here that if we express the number of plasma exchanges performed per 1 million inhabitants in 2014, you can see here the difference between countries, because there are countries that they perform more than 400 plasma exchanges per million, while there are some others, for instance, in the range of 150 or even lower. Also, for us, was a surprise to see Germany that you see that it's just below 200. Probably, remember, because in the central of Europe, most or an important part of plasma exchanges are performed by a nephrologist. And obviously, we do not collect data of them, okay? But that's just an indication of the different practices in the performing apheresis, but in this case, plasma exchange in different countries. What about side effects? This is just a reminder of the numbers that we found our colleagues found in 1980-85. Fortunately, we have improved. These are the data that we published when we analyzed more than 2,200 plasma exchanges performed in our institution between around 10 years. And you can see here that the percentage is of side effects when we use albumin, it's just about 3%. And most of them, they were just minor hypocalcemia and mild hemodynamic changes. So fortunately, with the measures that we have introduced to prevent those side effects, nowadays plasma exchange is a safe procedure and effective, and obviously zero transmission of infectious diseases when albumin is used, because it's already by the separation method is sterile, but also when we use plasma, because currently we are using pathogen inactivated plasma when we use plasma as a replacement solution. But also, we have learned that we have to combine plasma exchange with something. And this is just a good example. These are the data that Jim George presented at the meeting of the American Society of Hematology, of ASH, in 2012. Probably you know that Jim George in Oklahoma has probably the most important registry of thrombotic microangiopathies in the world. Well, they were analyzing the number of patients that were treated with plasma exchange due to TTP, an episode of thrombotic thrombocytopenic purpura. You can see here that in those years, 1996-99, they have 11 patients. Those patients required 27 days of plasma exchange. At that time, we didn't know exactly what was the cause of the TTP. So for now, it could be just surprising that just 55% of the patients were receiving immunosuppressive drugs, because, now we have identified that TTP is an autoimmune disease. So the plasma exchange, okay, we're removing the antibody, but if we do not inhibit the production, the antibody will continue, okay? But with the time, along the years, we learned that it was an autoimmune disease, and we learned that we had to treat with steroid, but also to introduce rituximab, because it is a much more efficient way to inhibit the production of the antibody. And look at what happened. In the 2000s, with just 8 days of plasma exchange, we had enough to induce the remission in the patient, compared to 27 days. And obviously, we were performing plasma exchange just 8 days. The number of side effects also decreased. Why? Because most of those patients were being treated using a central venous catheter. We will see later that this can be improved. So, meaning that just the importance of combining the plasma exchange with the inhibition of the production of the antibody. And this is just another example. This is the last published version of the Esfh guidelines, and referring to the autoimmune hemolytic anemia. The indication for cold agglutinin disease, is a disease produced by AGM, and the AGM is mainly intravascular. This is a category 2, and with a high degree of recommendation. But for the severe warm autoimmune hemolytic anemia, IgG, you see that the category is 3. Why? Because obviously the problem is that if we are removing the IgG but not inhibiting the production, we will see no response. But we published an experience with 2 patients with a really severe form of autoimmune hemolytic anemia. In this case, for instance, the nadir of the hemoglobin reached about 30 grams per liter, in spite of giving corticosteroid and giving intravenous immunoglobulin. But at that time, we decided to introduce rituximab, and we started to perform plasma exchange. And you can see here that in just probably in 1 week, we were able to remove the antibody because we saw an increase in the hemoglobin and a decrease in LDH as a marker of hemolysis. So remember that the indications of the plasma exchange might vary if you combine other treatments that help to inhibit the production of the antibody, so the efficiency of the plasma exchange is much higher. And this patient had a similar curve, where after waiting several days, we started to perform plasma exchange, and then the hemoglobin recovered again, and the LDH decreased. Well, let's try to talk about the future. Obviously, the future is very difficult, almost impossible to, but let's try to learn from the data that we have. And just as an example, this is the summary of the activity in our apheresis unit at the Hospital Clínic of Barcelona. We started collecting data in 2007, and almost 20 years ago, you see that the total of the apheresis procedures that we have performed has increased almost 400%. What else? We could say that along those years, we have been introducing new types of therapies. For instance, in, immunoabsorption, we started to use 4A bioincompatible transplant in 2008. LDL apheresis, we started to perform in 2011. This has been increasing along the years, and particularly extracorporeal photopheresis. Look at, we introduced photopheresis in 2012, and now we are in the range of 400 per year. And with a peak in 2023 that we performed more than 500. Because at that time we were performing a clinical trial, a randomized clinical trial about the role of ECP in the prevention of kidney rejection. And we use the Theracos technology. And you know, with Theracos, you have to perform 2 treatments per week. Well, areas of growth in apheresis. Indonesian probably is, because you know, is the highest activity that we have. Well, plasmapheresis. Why? Because Europe is heavily dependent on US plasma. Europe consumes 25% of the world's IgG, but only collects about 14% of the plasma for fractionation. Globally, so self-sufficiency of plasma-derived cerebral products is about 60%. We have to import products from US, 40%. It was estimated in 2020 that Europe, in that year, Europe would have needed 4.3 million liters of plasma more to meet the needs. Obviously, we didn't have it, so we were importing those liters from US. What about therapeutic apheresis? We see that, well, probably you remember that we were performing about 800 plasma exchanges, mainly in 2025, mainly because we increased our activity in hepatology and neurology. And for instance, it will be presented an abstract in the next European Association for Studying the Liver Congress that we will hold in Barcelona. We were comparing the plasma exchange in the treatment of the acute and chronic liver failure. This is really severe complication with a very high mortality. And we were looking at the role of the plasma exchange, combining albumin and plasma for treating those patients. It was a multi-center, a randomized controlled trial, where more than, the number of patients is somewhere here. 274 patients were randomized to receive standard treatment or plasma exchange, standard treatment plus plasma exchange. And what we found is that in the group treated with plasma exchange, the 90 days overall survival was about 35% higher in the group treated with the plasma exchange in comparison to the standard treatment. So you can deduce, you can think that probably when our colleagues in the pathology know that those data, probably they will start calling you to perform plasma exchange to treat this really common condition with high mortality that you can reduce significantly when you apply plasma exchange. More areas of growth. Extracorporeal photopheresis. This is something that is being used mainly for GVHD, but the experience in solid organ transplantation is very low. And there are now a strategy or studies running trying to look at what is the role of the photopheresis in solid organ transplantation. Because for instance, in our center, we have found that extracorporeal photopheresis has an immunogenic effect. Not immunosuppression, immunogenic, because we were treating some patients with kidney rejection with extracorporeal photopheresis. They responded, the rejection remained stable, but surprisingly, we saw a decrease in the virus, BK polyoma virus, that commonly infects the transplanted kidney. And with those, we saw that the replication decreased. So it is interesting. And now we are using ACP for treating patients with nephropathy provoked by the BK virus. Cellular therapies, particularly for hemoglobinopathies, you need between 12 and 20 millions per kilo of CD34. So it's a lot. So probably we will have to keep performing collections for treating those kind of patients. But probably the change, the biggest change that we will see in the future is CAR-T therapies. I'm sure that all of us have heard of the role of the CAR-T cells in malignant diseases. But please, see all the list of potential indication and studies that are being conducted nowadays to check, to investigate what is the role of CAR-T in autoimmune diseases, fibrosis diseases, senescence diseases, or even infectious diseases. However, I would like to share with you something that has just been published, because it was published in Nature Medicine just a couple of weeks ago, and some Chinese group was able to generate a CAR-T just injecting the construct into the patient without the need of performing a mononuclear cell collection from the patient. And they treated 5 heavily pretreated male patients with myeloma, and they found in 4 of them objective responses, including 3 stringent complete rejection. So perhaps in the future we will have a lot of CARs but they will not need us for connecting mononuclear cells because they will be able to inject directly the construct to the patient. And that's all. I would like to conclude that saying that apheresis donation and therapeutic procedure has not yet reached 100 years. In recent years, we have seen an increase in both the number and the type of procedures, and it is foreseeable that in the coming years, we will continue seeing growth in both the number and the complexity of the procedures. And that's all. Thank you. Thank you very much for your attention.
Thank you very much, Miguel, for this wonderful lecture. Since this is an award lecture, there will be no questions allowed. No questions allowed, unless you allow some questions. Then, this is highly unusual, but since we have time, we can have 1 or 2 questions for an award lecture. Now is your time to shine. So I have got one question. You also published a paper about plasmapheresis in TTP, and in the caplacizumab era, do you think that the plasmapheresis will still have a role in the future? Because in the past this was the game changer, and what is the future going to be in TTP?
Good question. Thank you, Andy. Yeah, that's true that nowadays the approach of treating autoimmune TTP has changed with the introduction with the arrival of caplacizumab. But we are convinced that we have still to perform plasma exchanges. Why? Because in our protocol, we have defined that we will continue the plasma exchange until the activity of the inhibitor is reduced in the patient and the activity is recovered. Why? Because if you stop plasma exchanges, obviously the antibody will continue circulating in the plasma of the patient. Remember that IgGs have a half-life of 3 weeks. So if you do not remove them, you will continue having in the circulation for at least 2 or 3 half-lives. That would represent probably between 6 to 9 weeks where you will have to give, to continue giving caplacizumab. And you know that caplacizumab is not a cheap drug. So with the approach that I have just explained in our center, the mean of treatment of the patients with caplacizumab is between 2 to 3 weeks. Why? Because we inhibit the production of the antibody with rituximab, and then we keep removing the antibody with plasma exchange until the activity has disappeared, and so the need of the days of caplacizumab treatment is reduced. So in my opinion, we will have to continue performing plasma exchange.
Thank you very much.
Thank you.
So if that was all, then thank you very much again and congratulations, and thank you for your wonderful contribution to the field of apheresis, Miguel. Thank you. So I hand over to the chairs of the next session.
Hi. Wherever you prefer. Sure. In the middle. Okay. Yeah, good afternoon, everybody. Welcome to the session on CAR T-cell therapies. And CAR T-cells has rapidly transformed the treatment landscape in hematological malignancies, and we're also seeing exciting developments beyond this field, as well as important advances in pediatric applications. And today's session will cover these three key areas: the established use in hematological diseases, but also the emerging indications beyond hematology and CAR T in children. Together with my co-chair, Sylvia, we're very pleased to guide you through this session. And let's begin with our first speaker. Shall I introduce our first speaker?
So the first speaker is Jan Vidra. He's the head of the clinical division at the Institute of Hematology for Blood Transfusion in Prague.
So thank you very much for the invitation to this conference. So my task is to give overview of CAR T-cell therapy. I am a hematologist, hemato-oncologist by profession. My main area of interest is stem cell transplantation and cell therapy. So I will be talking not so much about apheresis, but about what we do with the products that are actually started by apheresis. Is there a something to propel this forward? Oh, thank you. So at first I will talk about the general principles of CAR T-cell therapy. I'm sorry, this probably has been repeated many times, so I will go over this quite fast. Then I will talk a little bit about apheresis manufacturing, and then mainly delve on clinical results, approved indications, about toxicity and its management, about novel targets and some new indications in hemato-oncology, and then about next generation approaches, and try to provide some insight into what can be expected. So first of all, CAR T-cells are based on introduction of chimeric antigenic receptor into T lymphocytes. We also have CAR NK cells being developed now. So CAR is the interesting invention, is this, which is a chimeric protein. Chimera was a mythical monster composed of a body of the lion. It had also a head of a goat and a tail of a snake. Actually multiple snakes, so it was composed of parts of multiple creatures, the same way as the chimeric antigen receptor is. So it has a binding domain of a immunoglobulin, then a transmembrane domain from I don't know which protein actually. It has a co-stimulatory domain and a CD3 zeta chain which provides the transduction signals. There are several generations of CAR receptors. The first generation, the original inventions in 1990s was composed only of the immunoglobulin binding domain, transmembrane part, and a signaling domain. And then this didn't work very much because these cells didn't expand, they didn't persist, and they didn't have much clinical efficacy. So then another part was added to the chimera, which was the co-stimulatory domain, either 4-1BB or CD28. And this is actually a design, the second generation CAR T-cells that is used in all of the currently approved CAR T-cell products. Then there is a proposed third generation, which has two co-stimulatory domains, and a fourth generation, which also has either cytokines as part of the constructs. So these are called armored cells. And we are still waiting for these to go through clinical trials and maybe get into clinical practice. But the second generation is probably the balance between too much signaling and just enough signaling, because what happened with the third generation cells often is they had too much stimulations, and these T cells would actually exhaust themselves very quickly and did not persist. So we have all been living through this history, which started to be very, very public in 2012, where first complete remissions were reported in Emily Whitehead, who was the child at that time that was pretty much publicized her cure of acute leukemia. She is still in remission, doing fine, at least what I know. Then in 2017, the first approvals came for B acute lymphoblastic leukemia and then non-Hodgkin lymphomas. And then the indications expanded also to multiple myeloma. And now we have seven products which are approved and in clinical use. These are the list of products. Of course, in EU, the approval is always a little bit later than in the United States, as it happens in probably all areas of medicine. And the indications which are listed here have all one theme in common. It all starts with relapse refractory patients, and then the treatment is moving into earlier lines of therapy. So basically, we have a CD19 targeted therapies which are used for therapies of B acute lymphoblastic leukemia, diffuse large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma, and primary mediastinal B-cell lymphoma, and then BCMA targeted therapies for multiple myeloma. The access to these therapies in Europe is quite diverse. It's not uniform. This is a recent publication from Hemisphere, published this year, but the data are about 1 year old. And as we can see, the number of approved indications in different EU countries or available therapies in different EU countries is quite variable. This was a bit surprising for me. I thought that the EU would be more homogeneous in this regard, but that's just the way it is. So let's focus on the patient journey. It all starts with indication and then with leukapheresis, T-cell collection, then manufacturing, and at the same time that the cells are being produced, the treating physicians need to make sure that the patient actually makes it to treatment. So some patients, or most patients, do need some form of bridging therapy to control their disease before they actually receive the CAR T cells. Then they come in for low-dose chemotherapy, they get the infusion, we manage complications, and then there is the question of long-term follow-up and long-term complications, and maybe management of relapse. So leukapheresis, collection of starting material, is the first critical step. There have been some recommendations published on how to optimize and perform this, and I will not talk about it, as this is not my expertise really, and you are the experts in this field. What is important though is that there are risk factors for treatment failure, both of manufacturing failure and actually treatment failure, which is prior therapy with bendamustine, use of fludarabine just prior to T-cell collection, and advanced disease, high LDH, high ferritin, CRP. Generally, a progressive disease is a sign that things will go bad. Now, with regards to clinical results, it all started with pediatric BL, which I will not discuss too much because there will be another talk on pediatrics. However, similarly good results are reported in adult BL as well. So this is a real-world analysis of a registry of commercial CAR T-cell therapy for adult BL, mainly with Brexisol, and what's seen that there is high complete remission rate. There is high rate of even MRD negative CRs. However, patients still do relapse after this treatment. So 1 year progression-free survival is about 50%. And the 1 year overall survival is 60%. So this is quite similar to the pediatric data, and what it tells us that this is a very advanced and successful treatment. The situation used to be much worse for the patients, but this story is not finished, not nearly at all, but unless we are able to cure a real majority of patients, then we are not finished with this work. So I think that this is an important step, but we are halfway through. Now, when this CAR T-cell therapy was introduced, what we thought that it would replace allogeneic stem cell transplantation, maybe even for patients with acute lymphoblastic leukemia. This probably is not the case, and it seems that at least for patients who received CAR T-cells which have CD28 co-stimulatory domain, which do not persist for that long, these patients still, if they have B-ALL, may benefit from hematopoietic stem cell transplantation as a consolidation of their treatment, especially if they have not undergone transplant before CAR T cell therapy, if this is their first or second line treatment. So there are no randomized data to support this. This is an expert opinion, but that's the situation how it is now. Especially patients who have high-risk disease or MRD positive disease still require hematopoietic stem cell transplantation after CAR T therapy. Now moving to non-Hodgkin lymphomas. The success story in diffuse large B-cell lymphoma is quite dramatic. However, the results are a little bit worse than what we see in acute lymphoblastic leukemia. And what has been the latest important step is the movement of this into second line from the original third line and beyond, where two pivotal trials, ZUMA-7 and Transform with Axicel and Liso-cel, show superiority to standard therapy, in contrast to Kymriah, which surprisingly did not show superiority in second line to standard therapy. Now, many discussions have been made about why Kymriah didn't make it through on this bar. It may be because of the design of the study itself, because of different definitions of endpoints. It may be because it is a less potent product. It may be because the production of Tisa cell took longer on that study than what is usual in current clinical practice. We will actually never know. It is what it is, and now we have these two other products which are indicated in second line as well with these results. Now the important, of course, impact to apheresis practice will be these patients would previously received autologous stem cell transplantation. Now they are treated preferentially with CAR T cells if they have relapsed within 1 year after therapy or if they are primary refractory. Now, another interesting topic is, are CAR T cells actually better than bispecific antibodies, or are they worse? Because the bispecific antibodies are off-the-shelf therapies. There are many reasons to prefer bispecifics actually against CAR T cells, while their mechanism of action is similar. Again, there are no randomized data in this regard, but there have been some meta-analysis of published trials produced, and it does seem that CAR T cells are more effective in treating these patients. And even though the bites are more convenient, this offers, the CAR T cells do offer a better chance of disease control with a single treatment. Again, a situation is similar with follicular lymphoma and mantle cell lymphoma, and I will not repeat much of this data. We have quite nice efficacy with some toxicity which is always present. Now moving on to multiple myeloma, we have two products now available which target BCMA. Again, with very interesting response rates. The indications are not equivalent at this point. The Carvykti is already indicated in second line and above. Abecma after failure of 2 lines of previous therapy. Although they both do target BCMA, there are differences in the binding domain, so they have different activity, and it does seem that the cilta-cel has a more profound clinical activity, but also a high risk of cytokine release syndrome, and it has a unique side effect of neurotoxicity resembling Parkinson's disease, which is not present in other forms of CAR therapy. So going to the toxicity of this treatment, we now know about the efficacy, and now the other part is toxicity. So when this gene therapy came in, at least to Czech Republic in 2019, we thought that it would move to first line closer and we would get rid of these nasty side effects of chemotherapy. Well, the side effects of CAR T cells may be quite as nasty as side effects of chemotherapy, so I wouldn't necessarily say that CAR T is always easy. The ICANS, neurotoxicity associated with CAR T cells, can be quite unpleasant and drastic for patients as well. So that's something to definitely improve on. So first of all, and most common, is the cytokine release syndrome, which is related to the biological activity of CAR T cells themselves. They do produce cytokines the same way as they would if they fight infectious disease. They produce IL-1, IL-6, so the patients do get the normal symptoms of fever, and then may develop multi-organ dysfunction and multi-organ failure as well, if left untreated. The overall incidence is a little bit under 50%. Most of the CRS are mild. At least in our center and in other Czech centers as well, we use prophylactic anakinra in all patients, so the rate of CRS has gone down quite a bit, and I would say that this complication is quite manageable at this point of time. ICANS is a little bit more unpleasant. It is more common with some products. For example, Axicel, in contrast to Tysacel, does lead to a higher incidence of neurotoxicity, and again, something that is quite unpleasant for patients, although it is manageable and it is reversible. I did mention the Cilta-cel has a little bit different profile of neurotoxicity, and that's something that we still will need to learn more about and work on. Management of both is based on anti-cytokine drugs with mainly tocilizumab or anakinra on top of the corticosteroids, dexamethasone or high-dose methylprednisolone. There are other toxicities which are not as common. One of them is immune cell effector associated cytopenias. Some patients do have prolonged neutropenia, thrombocytopenia, or anemia. Again, we can support them with growth factors, and that's about what we can do with this. Most patients will recover, but there are rare cases where this is quite unpleasant and annoying as well. Some patients may, and sometimes it may be difficult to distinguish this from underlying secondary myelodysplastic syndrome from previous therapies that the patient may have received before CAR T cells. The other B-cell aplasia, hypogammaglobulinemia are less severe and something that we sort of may work around and do IgG replacement if necessary, and the patients do fare quite well actually. Something which also was a little bit unexpected is the rate of CMV reactivation, which does happen in patients after CAR T cells. It's something that we didn't think at the beginning of the program would be a problem. It's not a huge problem, but it does happen. There is, from the beginning of CAR T cells, there is always a issue of a fear of secondary malignancies. Anytime that there is a genetic manipulation, there is always a risk that we would introduce genetic changes into these cells. Luckily, this is quite rare in these large cohort studies. Only single cases of T-cell lymphoma have been reported. And most of the other secondary malignancies were not deemed to be related to the gene therapy itself, but to maybe previous chemotherapy that the patients have received, like is the case with the secondary myelodysplastic syndrome. So moving on to modern indication, I will not list all of the applications that are being considered, just several model cases. One is CAR Ts for acute myeloid leukemia, something that would be very interesting for hemato-oncologists because it's a disease where we do not have really seen a significant breakthrough apart from venetoclax addition to azacitidine in older patients. So CAR Ts for acute myeloid leukemia, they do not work as well as in lymphoid malignancies. This is because there is not a good antigen like CD19 or BCMA, which could be targeted safely and eliminated safely. The antigens that are they are commonly expressed on AML cells, are often shared with other myeloid progenitors and with hematopoietic stem cells. So there is always a fear of inducing cytopenia or bone marrow aplasia. Also, there is a problem of immunosuppressive tumor microenvironment. The AML blast cells probably produce cytokines that limit the activity of T cells. And also there is a microenvironment in bone marrow which does play a role in this as well. So the response rates are very low. There is a short peak activity. The cells are difficult to produce. They don't expand, and some trials are listed here. And in general, with current approaches in AML, the response rates have been generally very, very low and disappointing, and persistence limited, and also high rate of manufacturing failure has been always seen. So there will be there are several new strategies that are being tried in AML, but also in other CAR T therapies. One is to use logic-gated CAR T cells. That's something that's very interesting. Actually having different chimeric antigenic receptors, some of them activating, some of them inhibitory, and making these cells more intelligent so that they do not rely on a single antigen present on the target cells, but they actually are able to distinguish disease cells from healthy cells using a combination of factors. There may be a need for an approach where this therapy is combined with stem cell transplant with gene-edited cells, so that we can delete the original hematopoiesis using CAR T cells and give the patient back hematopoietic progenitors which would be resistant to CAR T cells. Other approaches would be to use adapter CAR T cells. One of them are called unicars. That's a very interesting approach where these CAR T cells are aimed against an antigen that is not normally present on cells, and at the same time the patients are infused a linker protein which actually does the binding of these CAR T cells to the target tissues. And this is an important or useful idea to improve safety, because then these cells can be safely turned on and turned off, and actually turned on and off. CAR T cells for T cell malignancies would be very useful, because T cell acute lymphoblastic leukemia is a huge clinical problem, although it's quite rare. The cases which are resistant to therapy are just horrible to manage. So these CAR T cells, again, their production from autologous cells is difficult, because the T cells will kill the T cells, which is called a fratricide. For example, if we modify these patients' T cells to target CD7 or CD5, they will kill themselves in the culture. So approaches are needed to either knock out the original CD7 or CD5 in these cells, or to use allogeneic cells in this to produce them. So there are several other approaches that are being actively investigated. Is production of allogeneic off-the-shelf CAR T cells, which would bypass many problems with especially patients like AML, where there is not enough time to actually manufacture the T cells, the CAR T cells from them. Now this is a technologically much more complicated issue than just producing the conventional autologous CAR T cells, because on top of adding the CAR receptor itself, the manufacturer has to knock out the original T cell receptor. Maybe they also need to knock out HLA genes to avoid immediate rejection. And this is a vastly more difficult technology than simple lentiviral transduction summarized at this slide. So there are many things being tried, and I think that the general incentive of having an off-the-shelf CAR T therapeutic is very huge. It would be highly practical to be able to just take this product from the biobank or from the pharmacy, and not have to go through all the individual's logistical process. So maybe the incentive is there, and maybe this is where the field will move, but we will see. Another approach that is being tried is in vivo CAR T-cell generation, which was already discussed. Basically, the lentiviral vector is infused into the patients, and the cells are produced in vivo. So this is something that also is very interesting. And there are several other approaches, which I will not go into more detail because I think it's been quite long already. So there are several challenges which will need to be tackled. One is clinical. The patients do relapse, so there is a risk of antigen escape. If we target in single antigens, the cells will lose the expressions of the antigen. They will exhaust themselves. So there is a question of optimal sequences of these therapies. So there is lots of clinical issues to look at with the current generation, but more likely we will need more advanced therapies to actually move forward. There is logistical problems with getting slots for apheresis, which can become a bottleneck in this whole process. Centralized manufacturing is always costly, and it is a problem. For some patients, the waiting time is an issue, and of course, the limited availability of treatment centers and expertise may be an issue. Economic problem is clear. This therapy is very expensive and will be for some time, unless there is some change in technology like decentralized manufacturing or point of care manufacturing, which is actually currently possible, and which I think is a very interesting possibility of future development. As I mentioned, there will be, in order to make this really a universal treatment approach, lots of science needs to be done. Approaches do need to be invented to make these cells more intelligent, not to just be on-off and target single antigen, but to really be able to recognize different cells. So we'll see how things work out. At the current point, I think that we cannot be sure. I would predict that we will need many more apheresis centers, because I do think that the field will evolve into multiple more indications. So I do believe that we will need more treatment beds and more apheresis centers, because I think that for maybe next 10 years we will still be doing lots of autologous CAR T therapies. But we will see. There is still a possibility that someone will come with a radically different approach, and we will all move to in vivo, single shot, no apheresis needed scenario. But I think that that's not the case for the next 10 years. So thank you very much for your attention, and thank you.
Thank you very much for this very, very good review about CAR T cells. Do you have any questions in the audience? So maybe if I can start with one question, if you don't mind. As Prof. Lozano mentioned before, in Europe consumes 25% of immunoglobulin of the world, whereas we only collect 14%, so we are not self-sufficient. And one of the main complications is the hypogammaglobulinemia. So with the CAR T cells, there's an increase of need for immunoglobulins, because we replace every 3 months, more or less, no? What do you think is the future if we don't have enough immunoglobulins?
So interestingly, quite often the patients, adult patients, after CAR T cell therapy, they do not always require immunoglobulin supplementations. Some of them do. So we do check the levels, and then some of them will need them. I would say that Europe is not self-sufficient in many areas, which now seem like a problem. Not only immunoglobulins, we will probably have to work on this as well. Yeah.
Any other question? We have 5 more minutes.
Okay, thank you very much. Then I would like to go to our next speaker. Which is Julio Delgado. Is working in a hospital clinic in the University of Barcelona in Spain, and he's going to talk about other uses beyond hematological malignancies.
Okay, well, thank you very much, Silvia and Claudia, for your kind introduction, and thank you to Gerda and all the organizers for inviting me. This is my second time at this meeting, so I'm very pleased to be back. I changed a little bit the title because actually what is a problem is not really beyond hematology. What is new is not only beyond hematological, but actually beyond B-cell malignancies, because all the products we have and we currently use are all targeting B-cell malignancies. All the other malignancies are left behind. So it's going to overlap a bit with the previous, but only a bit, so don't worry too much. So these are my conflicts of interest. I don't have any from pharmaceutical companies, but I am a worker of hospital clinic, and I'll mention briefly 2 products that we are manufacturing in our own institution. And of course, you know, I'm an employee of the institution. My salary doesn't change depending on how many products we infuse, unfortunately. But you could see this as a bias. I'm sure you could think that this is a potential bias. So there's no need to discuss how important CAR T cells are. I mean, these are the 7 products currently available in the US and in the European Union. And yes, all the indications are B-cell malignancies. And there are several reasons for this, but I'm just going to go back a bit and try to discuss with you why these products are so successful. So the first great idea is a very old one, and it comes from the 90s, actually end of the 80s and beginning of the 90s. And it was these Israeli researchers who got this amazing idea of combining a monoclonal antibody with a T-cell machinery, all in one molecule. And why is this so important? Because monoclonal antibodies are able to identify antigens in any way, soluble, attached to proteins. They are HLA independent, whereas TCRs, the natural receptors of the T cells, they only recognize antigens that are processed and presented by professional cells within the HLA groove. So that creates a problem when you are developing a TCR, because TCRs are HLA dependent. So you have to develop a TCR specifically for each patient's HLA if you want that TCR to be useful. CAR T cells, well, no, not at all. You don't care about the patient's HLA. You just give the cells, and you will use the single chain variable fragment of the immunoglobulin to identify the antigen, and the T cells will get rid of the tumor cells. So again, that was done back in the 90s. The second very important achievement was in the late 90s and was second generation CAR T cells. My predecessor just mentioned it, that all the approved products are second generation CAR T cells, because first generation CAR T cells did not work. So when you use one signaling domain, that cell doesn't work. And instead of giving up, you know, the researchers, all these academic researchers, start thinking about ways of improving it, and they got to the second generation. And that is like magic, because nowadays these cells do work. And finally, and even more importantly, is culture technology, because these cells are happy when they are within the body, but not really happy when they're in a machine. So it's important for the culture to be short. If a culture lasts for 3 weeks, by the time you give them back to the patient, they're so exhausted they just die. So it's important to keep the culture short. And that is not an easy task. You need a lot of technology there. So you need a lot of cytokines, and other substances so that this culture is short. And that was beginning of the 21st century. And what is really, really important is that, we can live without normal B cells. So we don't really care if we kill all malignant and benign B cells. So that is why B cell malignancies are actually where these products are pretty useful, because we don't care too much about normal B cells. I have to be honest with you, this is true, but not really true. And I'm saying this because we now have patients in our clinic who've been in B-cell aplasia for 5 years, 6 years, and they're not doing that well. Even with immunoglobulins, they are not doing that well. So that mantra we keep repeating about it doesn't matter, it does matter a bit. I mean, it's not like you die if you don't have normal B cells, but your life isn't easy if you have prolonged B-cell aplasia. So you see, when we put this slide that we put all the time, this is actually a summary of 20 years of amazing progress. Yes, it looks like simple, but it's actually quite complex, and it took a lot of work and effort to get to this magic circle where we collect the cells, we transduce them, we culture them for a short time, and then we give them back. So because killing B cells is easy, the easy indications to come are autoimmune diseases, because of course patients with autoimmune diseases have abnormal B-cell immunity together with T-cell immunity, so no wonder several academic and pharmaceutical companies have developed products to treat autoimmune diseases. And the results are actually quite striking, even more than you would have expected. Because you see, we've been using rituximab to treat these diseases for a number of years, and the results are not the same. This is one study that was published late last year, and you see here results in patients with SLE, with lupus, and the responses are actually really, really good, and the signs, the clinical burden of these patients goes down quite quickly. Here are the results of patients with scleroderma or systemic sclerosis. Here it's more difficult because there's a lot of fibrotic tissues there, so patients do not improve that much, but they do improve. When you ask them, this is how they feel, and they actually improve somehow in terms of their symptoms, and they also improve in some things like DLCO, etc. So pulmonary function, etc. improves. And back here you have the results in patients with inflammatory myositis. They also improve a lot with the treatment of CAR T cells. So again, quite interesting data, and you know very well, lots of clinical trials going on in all our centers with a variety of autoimmune diseases. But even neurological immune diseases are now potential candidates for CAR T cell therapy. We are already participating in clinical trials as well in patients with multiple sclerosis. In this case, it's a BCMA construct, the paper I selected for you, but CD19 CARs have also been used to treat multiple sclerosis. This is one of the few actual papers that are published in Cell. There's 5 patients with multiple sclerosis here, and this is a type of test they usually have, and well, there is an improvement in that type of test that is actually walking for a patient with multiple sclerosis. What you see also is the expansion of the CAR T cells, and it's interesting that the expansion in these patients with immune diseases is typically shorter. So we don't care that much about CAR T cells persistent in patients with autoimmune diseases. We're just looking for an immune reset. So we just want to reset the whole immune system, and with that is usually enough to achieve clinical improvement. You don't actually need the CAR T cells to live for years and years, which is not the case for B-cell malignancies, I have to say. In our hands, there's a clear correlation between CAR T cell persistence and, for instance, progression-free survival in patients with acute lymphoblastic leukemia. So when you have a malignant disease, then it is better if your CAR T cells persist. For autoimmune diseases, maybe not the case. But even myasthenia gravis, another paper recently published, actually this month, shows that CAR T cells are better than placebo. So again, these are now doing randomized trials. Can you imagine? So you have patients with severe myasthenia gravis, and you randomize them to have CAR T cells or standard of care. And in these randomized trials, of course, there's not huge numbers, but still 20 and 18 patients is quite a lot. And you see there's a significant difference for patients receiving CAR T cells. So again, autoimmune diseases, neurological autoimmune diseases, of course, these are all autologous CAR T cells, lots of apheresis done. This is the part I overlap. I'm going to talk briefly about T-cell malignancies, just to say that this is not so easy. T-cell aplasia is a problem, fratricide is a problem, but there are products out there, and one is targeting CD7. These are results published recently, well, not recently, a couple of years ago, but I just wanted to say that we, as everybody, we are also developing our own anti-CD7 product, and we got funding for this, actually twice, and we are starting the trial hopefully towards the end of this year. The approach we're going to follow with this product is because, of course, CD7 is expressed by normal T cells as well. So what you do, because you put the construct in the T cell, and the T cell is going to recognize the other T cell in front of him that expresses CD7 as well, and it's going to kill the T cell in culture. So what we do is we select CD7 negative normal T cells. I wasn't aware these cells exist, but they do exist. So a small proportion of our own normal T cells are CD7 negative. So what we do is we select CD7 negative T cells, and we prepare the CAR T cells on those cells so they don't kill each other. We don't know what's going to happen because we haven't treated any single patient yet, but we are looking forward to it. AML, another very difficult one, very heterogeneous disease, and I think this slide shows it all. I mean, look at all the different targets that have been tried. Whenever you see there's lots of targets that have been tried, that means none of them works. So really, really difficult disease to treat with CAR T cells. And something that is quite extraordinary, many groups believe that for AML, NK cells are better. There's no clear explanation for that, but it looks like this is the case. So some groups are actually working not on T cells, but NK cells for treating acute myeloid leukemia. We'll see what happens. And I'm also looking forward to see any product doing any good to AML patients. But now comes the very difficult part, which is solid tumors. Oh my God, this is really difficult. Why? Well, you know, the tumor is heterogeneous. I mean, it's not like AML where 99% of the tumor cells express CD19 and yeah, we're all happy. No, I mean, that doesn't happen at all. So there's a lot of heterogeneity. That means there's a lot of antigen escape. So you're basically selecting the cells that are negative or have low expression of the antigen, and well, you know, they grow and blah, blah. Then you have a very hostile immunosuppressive tumor microenvironment that doesn't exist for hematological malignancies. And you actually have physical barriers because tumors usually have a capsule, these solid tumors, fibrotic capsule, and it's hard for these T cells to penetrate this capsule. So, wow, very, very difficult. So we need to identify suitable targets because the other big problem is that, well, there's hardly any tumor-specific antigen out there. And at the same time, we have to prevent tumor escape and reduce toxicity. So you see, very, very difficult task. But don't worry, I mean, there's so many brilliant researchers out there working on solid tumors that will see, for sure, approved CAR T-cell products for the treatment of solid tumors. Again, many antigens tested, few successes. And I'm going to show you some of the things that are out there. So this is actually what we are trying. So HER2 is well known. It's expressed not only on breast cancer, but also other types of malignancies like gastric carcinoma. So it's a good target. The problem is that HER2 is expressed in many cells, many normal cells, but at a low intensity. So when we talk about HER2 positive breast cancer, what we mean is overexpression of HER2 compared to normal, but not expression of HER2 compared to zero expression. So if you design a CAR T cell that identifies every single HER2 positive cell, the patient will die irremediably because you'll kill lots of endothelial, epithelial cells as well. So what people are doing and we are doing at our institution is we have designed a product that only targets HER2 high cells and not HER2 low cells. So we have these mouse models where we put a HER2 high tumor on one side and a HER2 low tumor on the other side. And success for us is killing the HER2 high without eradicating the HER2 low because we are concerned about toxicity. And this is something we're going to test also very soon. This will be our first product for solid tumors. We'll see what happens again. Very difficult. We don't know. But see, there was already a randomized trial, a positive trial in solid tumors. It was mostly gastric carcinoma. The target is called Claudin 18.2. And this was a randomized trial. And even though it's a positive trial, you know, I mean, the results are not great. I mean, yeah, I mean, there's some improvement. Yeah, there's some reasons to be happy. But look at the positive. I mean, this is the good curve. Not great. So there's still a lot of work to do. So I've selected a few new things that are, I believe, very interesting. First one, the concept of armor CARs. So you see, CAR T cells can be done by us. We don't need pharma to do them. So that's why it's so interesting. That's why so many advances, because we can do our stuff in our own lab. So this group, what they are doing is, instead of target this antigen here called GPC3 only, they put another vector and they co-transduce with two vectors. The second one includes an interleukin, interleukin 15. So what happens is these CAR T cells not only identify the tumor cell, but they secrete locally IL-15 that helps themselves to kill the tumor cell. So this group tried in patients the CAR T alone, and you see that in the swimmer plot that there were basically no responses, and with the IL-15, and you see here there are some responses, so at least one partial response or even two partial responses in patients with intractable solid tumors. So these are the type of ideas that are coming forward to improve the efficacy of CAR T cells in solid tumors. And this is even more interesting. This comes from New York. That's Michel Sadelin. He used to be at Memorial, but is now at Columbia. And look at this. I've shown you before the CAR T cell construct. The problem with the CAR T cell is that it's quite artificial. Because, well, this is a monoclonal antibody. This is a fragment of a monoclonal antibody. That means the immune synapse that you achieve is not great. That means you need a lot of antigen on the tumor cell, because the immune synapse is not great. So what he did is, using the TCR, which is the natural receptor for the T cell, and he only changed that bit here, the external part, and put the monoclonal here. So the rest of the molecule is exactly the same as for the TCR. There's only a minor change. But this change is needed so that this TCR is HLA independent. Because again, if you use a TCR, TCRs are not HLA independent. So you need a small portion of a monoclonal antibody to make it HLA independent. And yet you keep the T cell machinery as it should be. With this, they get higher sensitivity. So you don't need a lot of antigen on the tumor cells. With low antigen, with ultra low antigen, it works. And this is preclinical model, but this is in pancreatic cancer. And you see that this CAR T that targets CD70, there's a very interesting antigen here. When you add some other costimulatory domains, this HLA-independent TCR works really, really well. We're looking forward to the clinical trial. Of course, sometimes things that work in the lab don't work in the patient, but I think it's a very, very interesting concept. And of course, this is an apheresis meeting. In vivo products are like, well, not to mention. But I actually found a spin to it because, yeah, I mean, they are working on in vivo products, and they are potentially useful for autoimmune diseases. And why? Because these in vivo products do not persist. And I said before, for malignancies, I'd rather have a CAR T cell that persists. For the autoimmunes, it doesn't matter. So we're okay. The second thing is that for in vivo products, you may not need lymphodepleting chemotherapy. And of course, we hematologists, we don't care much about chemo because our patients have received tons of chemo by the time they come to us. So it's, I mean, what is going to do? I mean, a bit of cyclophosphamide is not going to hurt you. You've received tons of it before. But a patient with autoimmune disease is different. He has never received chemo. So for that patient, if he can't avoid that, is better. So this is the two things that may work for these in vivo products. The Chinese already use these lipid nanoparticles to treat some patients, and it apparently works. But a word of caution, and I've used the same paper Dr. Lozano used it, but to show the important toxicity that they saw in these patients with multiple myeloma. Very severe. All patients had grade 3 or higher CRS, and one actually died of toxicity. And what is interesting is that this trial was stopped after 5 patients. And if this was stopped, that means that, well, perhaps it's not such a great idea in vivo products for malignancies. So you can breathe for a while. Just concluding, nobody, not even transplant specialists, dispute the increasing value of CAR T cells. The potential is unlimited. And this is because it's not only pharma business. It's not only for them to do this stuff. We can do this stuff as well. And that's the reason why this advances so fast. There are other indications like infections, prevention of graft rejection, things I didn't want to touch because it was just too much. But there's so many things you can think of it can be used to, CAR T cells can be used for that, well, stay tuned because new developments will blow your mind. Thank you very much.
Thank you very much for this amazing overview. I think we have time for questions. Are there any questions?
Yes, sure. And Nina's going to ask me a question too. Yeah.
You first, Nina, or? Okay. Dr. Delgado, really, really, really amazing presentation. Thank you so much. You spoke already about the acute toxicity, and we would like, or in fact, I don't know, it's a Dutch saying, we would like not to come from the rain into the drop. In other words, in the English version, it's out of the frying pan and getting into the fire. Mm-hmm. We have now more than 20 years experience in the in vivo use, and the first survivors of 20 years are 25 years are there. I know there is some fear of development, and for the first gene therapies we saw development of leukemias and these kind of things. What are the long-term effects of these kind of CAR T use, or maybe this is also
in vivo?
Yeah, in vivo.
Well, it's just starting, so it's impossible to know what's going to happen. But they do not persist. There was a joke. They do not persist, so they basically go away. I mean, they don't integrate that much, and they're actually designed not to do that. So I don't think that is going to be a problem. Well, I don't think. I also thought that was not going to be a problem for lentiviruses. But for in vivo, I'm not too much concerned. My concern is actually the persistence. Because for malignancies, and I'm a hematologist, I mostly work with patients who have malignancies, I do believe persistence is important. And these in vivo products, same as allogeneic products, by the way, allogeneic products, the results I've seen, the persistence is not as great as for autologous products. So for the time being, for malignancies, I would say autologous CAR T-cell products will remain for years. That's how I see it. For malignancies. For other newer things, myasthenia, blah, 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 yeah, that could be the goal for in vivo products. And here, of course, I believe pharma companies will do better because that's more the business, really, because they hate apheresis and cells going back and forth. I mean, it's not the business. They have never done it. They don't like it. They don't feel happy with it. So the autologous will remain in the hands of academia. Well, basically, I mean, we will all be doing our own CAR T cells, autologous, and the new things, like in vivo, they'll come from, I think it's going to be harder for us to compete with pharma for an in vivo product. It's going to be harder.
I gave the microphone to Dr.
Okay.
Thank you, Julio, for this amazing talk, really, very wonderful. So how far are these approaches that you infuse CARs and then exchangeably can use their target, the CAR, to switch on, switch off if it's needed?
Yeah, how far? Well, these are very, very nice concepts, but actually seeing them in patients, not that much. Again, because academia can do all this, and of course, if the goal is to publish a paper, then you do it, and you do this wonderful science, and blah, blah, and then you publish it, and you get recognition, and then they invite you to give talks, and then you get more money for grants, and blah, blah, blah. But if your goal is to treat patients and develop products, that's a different matter, because that is It requires a different set of skills, and many of these new designs are abandoned as soon as the paper is published. Because basically the goal was to publish the paper, not really. So that is something that we need to improve on. Because we, as a scientific community, we are normally interested in publishing and then getting grants and then more publishing and more grants and getting invited to conferences.
But that's the problem. You have a lot of phase 1 studies which never ever end in a product.
Exactly. That is the problem. So let's change the way we face this problem. Let's think that we are capable of doing stuff on our own. And let's do it. So you have a fancy design, you think it's useful, let's work, let's do it. I mean, you don't need to find somebody to do it for you. I mean, you can do it yourself.
Wonderful. Thank you very much for this talk. I have a question. I'm a pediatrician. And what we saw in our CAR T-cell population is that the success in total is not that what we thought it should be. We are lucky that Mrs. Whitehead, meanwhile, is still alive, but roughly 50 to 60% of our patients are not still alive due to escape mechanisms. And so we are asking ourselves, might be CAR T-cells are a good consolidation, but a cure not, because we have these mechanisms of the leukemic stem cells to escape this therapy. And what's about in lymphomas and other treatments to consolidate this with an allogenic transplantation?
Yes, I mean, ALL is a particularly difficult disease, and for children and for adults even more. So that question is always there, and whenever we have a patient who receives CAR T cells who has never had a transplant before, we generally recommend a transplant. So I know it's debatable, and some people would say, well, you gave the patient a 4-1BB construct and is still in B-cell aplasia, you could wait. But the fact is that approved products for adults are CD28. There's no 4-1BB product. Our own product is 4-1BB, and the median duration in adult ALL is not as long as it is for patients with CLL or follicular lymphoma. So we do see lots of relapses as well in adult patients. And some of them are actually CD19 positive because they have lost the CAR T cells by the time they relapse. So we do recommend transplant. These are not easy transplants, though. And our transplant colleagues are not happy when we say, yeah, now the patient is in fantastic remission, MRD negative, B-cell aplasia, do the transplant. And they run into herpes 6, whatever, I mean, lots of infections. So not easy transplant for them, but this is still something we recommend. But then for other diseases, it's debatable. For CLL, follicular lymphoma, or even DLBCL, I wouldn't recommend it normally. And then there's the other issue, which is that almost 90% of the patients we receive for CAR Ts have already failed an allogeneic transplant. So the question for us is usually, should we recommend a second transplant or not, rather than first or not? If it's first, we would normally recommend the transplant. When it's a second transplant, it's going to be reduced intensity by definition, regardless of a patient's age, unless you change the donor. I mean, it's a difficult discussion.
Yeah, I think we have time for 2 quick questions.
Quick question. Julio, thank you very much for this wonderful presentation. So do not be surprised if you are invited a third time.
Okay.
My question is, what could we do for avoiding the antigen escape that you have mentioned in 30-40% of patients?
Well, I wish I had an answer, but in solid tumors is a big problem. So there are many things that can be done, like targeting two antigens. The typical replies to that question are targeting two antigens. But another one is these armor CARs, because what happens is that when these T cells are there in the vicinity and there's a lot of cytokines, then other T cells, other immune cells are activated that perhaps may kill these antigen low cells. So you see, having these armor products could be the solution. And then other ideas like the HIT. I mean, the model that Michel Sadelain used for this science paper included CD70 low and CD70 high cells. And what he demonstrated with this HLA-independent TCRs, the sensitivity is so high that it doesn't matter. I mean, you kill patients, tumor cells that have low antigen expression as well. So it's just for normal CAR Ts that you need a lot of expression. For these newer versions, you may not. So, but yeah, and the other part of the question is what you do when they send you a patient and you find a 10% CD19 negative clone, and you have already prepared the product. I mean, it's a problem we face normally, and we don't know what to do.
So congratulations to very nice talk, and thank you because the Spanish program of academic CAR T-cells is really a something for as a role model for us as well. We try to produce our own CAR T-cells as well. We have 2 phase 1 trials with academic CAR T-cells running, and we always say, okay, in Spain they can do it, so maybe we can do it as well. And one thing that I wanted to ask you is, how do you navigate the patent issue? Because our idea is that as far as we are in phase of trials and academia and we just play, it's okay. But then whenever we would have something that would really work, things may get nasty. So how do you deal with this?
Yeah, I mean, we basically don't deal with it. I mean, you see, when we started, we thought about all that. And our first instinct was to do nothing, because you say, well, there's patent issues, then it doesn't work, maybe it's too toxic, maybe it's too expensive, maybe blah, blah. I mean, and you start thinking about all the potential things that can go wrong. And, but then we said, no, let's do it. So yes, this could happen. I mean, there could be patent issues, there could be problems moving forward. Gilead may decide to recruit lawyers to close down our program. This could happen, yes, of course. But you see, we've already treated 700 patients, and nobody's going to take that away from us. That is done. So I would just recommend you to do stuff instead of worrying about what could happen in the future. If your question is, are you sure you have freedom to operate? My answer is no, I'm not sure. And actually, if you ask 4 lawyers and you present the data, you'll have 4 different opinions. And it's not easy, not even for them. So if it's not easy for them, I mean, you just do stuff and then worry about all that in the future. I don't know, unless you want to make money out of it, unless your goal is to become rich, then freedom to operate is important. But if your goal is to treat patients, just do it, like Nike.
Just do it. Thank you very much. Then I would like to continue with our final speaker of this session, which is Friso Kalkoen. He's joining us from the Netherlands. So we see him online, and he's working at the Prinses Maxima Center of Pediatric Oncology in Utrecht, the Netherlands. Welcome, Friso, and the floor is yours.
Yes, thanks for the kind introduction, and my apologies for not being in Prague in person. I would have loved to join your meeting, but unfortunately it was not possible due to duties on the ward. I hope you can see my slide and you can hear me, and please interrupt if not. So indeed, my name is Friso Kalkoen. I'm a pediatric oncologist in the stem cell transplantation and cellular therapy ward in the Prinses Maxima Center. I have no disclosures, and I will show data of our work, but not related to any pharma company I'm involved in. Indeed, the CAR-T story started in the 80s and thereafter in the beginning of the 2000s, with actually pediatric studies ongoing with CARs and first generation CARs. So no inclusion of a second co-stimulatory or a co-stimulatory molecule. And these studies were primarily done in solid tumors. But with the CD19 CARs moving towards chronic lymphatic leukemia, the option came to treat children with CARs. And indeed, the first patient was treated with a CD19 4-1BB based CAR developed in the Children's Hospital of Philadelphia, together with the adult hematologist and the immunologist in Philadelphia. And the first patient was treated. She actually experienced very, very severe cytokine release syndrome, and shown on the top left, with very high temperatures and also signs of disappearance of leukemia by a decline in LDH. Her cytokine profile was immediately monitored, and the immunologist and IC doctors were consulted, and this was the first patient treated with tocilizumab, a drug, an anti-IL-6 receptor antagonist, now registered for cytokine release syndrome, as previously discussed. There's a nice comment in the New England Journal of Medicine looking back at this case, and they interviewed Carl June, and how important the case of Emily Whitehead was, and how important the collaboration was to deal with these toxicities in these first patients, mentioned by Steve Grubb. Fortunately, this patient is still alive and actually studying medicine in the University of Philadelphia at this moment. So indeed, the options are endless, but indeed, there's also been a lot of hurdles. The second pediatric patient already showed that despite a previous excellent response, you can develop a CD19 negative relapse. So antigen escape was already shown in the second patient who could not be rescued. Fortunately, the story continued, and as we know, Kymriah was registered based on the Eliana trial, an international trial run by Novartis, and it led to the registration for FDA and EMA in 2018. We moved from 7 academic centers to 1 pediatric oncology center in 2018 and started Kymriah treatment in 2019. So currently, Kymriah is indicated for first relapse after transplant, any relapse after transplant, or second or higher relapse, or refractory disease. That's the current indication since 2018, and unfortunately no alternative indications or products have been registered. There's a lot of real-world data demonstrating the 40 to 50% event-free survival, depending on the definition, looking at either MRD, so as a very low level of relapse, or full-blown relapse. But the general consensus is about 40 to 50% of event-free survival. There are a lot of risk factors defining the outcome of a relapse, but the known risk factors like site of relapse, cytogenetic risk factors or the number of relapses were not predictive in the current era. Recently, TP53 mutated ALL was shown to be to have a less favorable outcome with CARs, but all the other known risk factors are not useful anymore. What is useful is the persistence of CARs. In B-ALL, it's been clearly shown that persistence of at least 3, but preferably 6 months of CAR persistence improves your outcome, shown by this retrospective analysis in the US group, with a landmark analysis showing the outcome. So if you are still MRD negative at 3 months, and you lose your CARs, you still have a high risk of relapse. While if you have persistent CARs, your outcome is favorable. This was shown by the French group in 50 patients, and that this predisposes the loss of CARs predisposes to CD19 positive relapses and not to CD19 negative relapses. What's shown in literature, and what we also experienced, that if you reduce the blast counts prior to CAR infusion, you reduce the risk of CD19 negative relapse, but you do not reduce the risk of CD19 positive relapse, as this is predominantly affected by persistence of CARs. The early relapse after HSCT is also predictive for early loss of CARs. So if you have a very early relapse, you use these T cells for CAR production, you have less favorable CAR persistence. The other factor impacting the outcome is any detection of disease by molecular MRD. If you have any detectable disease at day 28 after CARs, you have an extremely high risk of relapse. So a lot of groups are looking into improving the current the cell therapies. For example, timing, subset selection. So for example, selecting naive cells, improving bridging. General consensus is to avoid immunotherapies like blinatumomab or targeted therapies like inotuzumab, but preferably use conventional chemotherapy. Of course, we need to control blast count, and novel CAR-T products are currently looked at. Of course, you can discuss HSCT indications. I'll come back to that. We have focused a lot on the lymphodepleting regimen. It was shown already in the 2000s for tumor-infiltrating lymphocyte infusions that prior to T-cell infusion, the administration of lymphodepleting chemotherapy improves the outcome, both by eliminating the cytokine sink, reducing your regulatory T cells, and enhancing the antigen-presenting cell activation. This was also shown in 2015 that trials, including lymphodepletion, had improved outcome, and all CAR T-cell therapies ever since have included lymphodepletion. Although this could be doubted for example, CNS tumors. Improving the intensity of the lymphodepleting regimen has also led to improved outcomes. So the addition of fludarabine to cyclophosphamide improved outcome both in adults and in children, although not significant in children, and that was shown to be associated with an improved CAR-T expansion when adding fludarabine. The US group in New York piloted an increased dose of cyclophosphamide, a toxic drug, especially for fertility. So we tried to reduce this, and although it was more effective and with improved CAR expansion, the clinical outcome was comparable. And with the toxicity of cyclophosphamide taken into account, they did not proceed with this high dose of cyclophosphamide. And in general, 1 gram per square meter is accepted in pediatric indications. What we did, this is the standard flu dosing regimen for Kymriah, so 4 times 30 milligrams per square meter. And based on our experience in stem cell transplantation with pharmacokinetics, we measured the actual flu exposure in patients on day 1 and 4 to define the exposure. And we saw a wide variation in the patients treated, ranging from 8 to 22 as an exposure to flu. And that was clearly associated with outcome. And so increased flu exposure, so more flu in the blood, gave a better event-free survival, and that was associated with less CD19 positive relapses and an improved CAR persistence, as shown by early B-cell recovery in the underexposed group. We also looked at cytokine profiles, and we clearly saw that cytokines like IL-7 and IL-15 were predominantly expressed in patients with a high cytokine, a high flu level, so more cytokines being available. We're currently looking into these in detail. And that indeed associated with an increased IL-7 and IL-15 being available over time. There are models to predict flu exposure based on renal function and body weight, and these were used by the US group to demonstrate in retrospective analysis that the predicted flu exposure is also related to clinical outcome, and they came to a similar AUC of 14. And that was independent of disease burden. They saw the predictive effect of the optimal flu exposure above 14. In line with this, different groups, both in New York and Spain, have looked at flu exposure in adult patients treated with non-Hodgkin lymphoma, and underexposed patients had a clear inferior outcome fitting the hypothesis. Although they saw more neurotoxicity, in particular CAR T cell related neurotoxicity like ICANS, so fitting with a more effective CAR expansion, but also the predictive effect of flu. The Spanish group demonstrated that you cannot fully predict flu on the individual level, so it's important to sample and know what you dose, and not just do that on a flu model. I don't go into this, but recently it was shown for multiple myeloma with a registered product that there's also an association with flu exposure and overexposure leading to CRS and ICANS with high predicted flu exposures. Now the question is, how can we improve? We can of course increase the dose, but that gives risk of overexposure and toxicity. You could use the PK model, but then you still have the disadvantage of potential overexposure, or you can do therapeutic drug monitoring. This is what we want to move forward, and we've currently opened an international randomized trial comparing the flu exposure with standard dosing towards increased dosing using higher exposure, starting with 40 milligrams per square meter instead of 30, and measure on the first day what the actual exposure is, and do TDM-based dosing on the subsequent days. This is a trial including 5 centers in Europe and 5 centers in the US, where we aim to recruit over 130 patients in a randomized fashion. That's as essential in pediatrics as numbers are limited, and we want to recruit this study within 2 years. Just to come back to the transplant discussion, we have previously published an algorithm who to transplant after CARs and who not to transplant, because taking into account the toxic effect of transplant, we defined a favorable CAR T outcome, so HSCT not particularly recommended, or unfavorable, and there's always a category where it's in discussion. So most important is B-cell aplasia or MRD negative when you become high risk, and CAR persistence and low disease burden are favorable. So I can refer to this paper if you want to look into this. And what you should take into account, can you salvage a patient with, for example, the novel drugs like blinatumomab and inotuzumab? Because now we give CARs mainly after transplant, but we see internationally that more and more patients receive CARs prior to allogeneic stem cell transplantation. But that is what we need to prove to expand the indication. We have some experience with patients prior to CARs, and we can salvage the majority, up till now, we can salvage all the patients we treated with CARs to prevent stem cell transplantation. To do this in a prospective way, Novartis opened a clinical trial in pediatrics for upfront treatment. Patients can be stratified to allogeneic stem cell transplantation after 10 weeks, so approximately 5 to 10% of the patients. And these patients allocated to stem cell transplantation were eligible for a CAR T product, the Novartis Kymriah product. They will eventually look at 4-year overall survival and 5-year disease-free survival. 121 patients have been infused, of which 6 in the Maxima, and we hope to see the first results this summer. A critical study where we hope to find ways to avoid the toxic effects of allogeneic stem cell transplantation. In line with that, and already mentioned by the previous speaker, and well known to everyone, the Barcelona product, or ARI-001, is the academic CD19 CAR 4-1BB based with a different binder compared to the commercial products currently available, using point-of-care manufacturing, and what we are working on together with the Hospital Clinic Barcelona, the Hospital San Juan de Deu in Barcelona, Paris, Rome, Ghent, and Utrecht will be trial sites who will also produce. So we will need to align apheresis, production, and release to allow for comparability. This study was funded by an attract call combination of funders from the respective countries that gave us the opportunity to run this large trial within Europe, with, of course, the challenge to align production between the centers. As mentioned before, some of the innovations come from pediatrics, and I like to highlight some of them in the coming slides. One of them is the challenge in TALL. Fratricide was already mentioned. The long-lasting T-cell aplasia after CARs against TALL is also a challenge, but can be salvaged by an allogeneic stem cell transplantation. And another aspect that might come into place in the coming years is product contamination, as you can, if you do an apheresis and harvest TALL cells, you can transuse TALL cells and they become CAR TALL cells, and therefore non-responsive to the CAR treatment anymore. So current protocols often include low blast counts at time of apheresis in these groups. In Europe, there's a CAR TALL study targeting CD7 actively enrolling, and that will be expanded to 4 trial sites, including the Maxima, in the coming years, where a unique concept is used to entrap the CD7 molecule on the CARs in the endoplasmatic reticulum. This has been published in a hospital exemption study. This hospital exemption program, and including 12 patients treated in study in Singapore, where they saw very deep remissions comparable to CD19 BALL CARs. leading to persistent remissions, but often requiring allogeneic stem cell transplantation as consolidation, and of course to prevent T-cell aplasia and toxic infections thereafter. There are major improvements in solid tumors in pediatrics. The group of Franco Locatelli in Rome published results on a third generation CAR, so including 2 co-stimulatory molecules with positive overall and event-free survival and long-lasting remissions after GD2 CAR T-cell infusion. They did not see GD2 negative relapses. What they saw is the effect of the microenvironment suppressing the CAR T cells by myeloid cells induced by the tumor microenvironment. And it was particularly shown that the patients that benefited were patients with extremely low tumor burden. Therefore, it's important in these novel studies to select and to be careful with your inclusion criteria, not to treat patients with too high tumor burden if you look at efficacy. Of course, you can do your phase 1 safety studies in these patients, but then you have to be aware that the curative intent is less. There are also novel studies focusing on GD2 in brain tumors, mainly by the Stanford group in the US, but similar studies are currently opening in London and Rome in Europe, and we hope to open a B7-H3 targeted CAR T-cell trial in the coming years for brain tumors, collaboration with London. There, the challenge is how to get into the brain, either by IV infusion or should you infuse right in the tumor. The Stanford group published remarkable responses, although the durability has not been shown, but of course improvement of neurological symptoms and MRIs is very promising. It comes with novel toxicities and severe toxicities, so it should be done in a trained team. Already mentioned, and I think here pediatrics lag behind by including autoimmune diseases. The first results are coming out, and we see similar effects. So great CAR expansion, but rapid recovery of B cells. This has been shown. This is also the Miltenyi product that's used here with point-of-care production. So the first publications are coming out, and I'm sure a lot will follow as the responses are comparable to adults, very promising with resolution of SLE symptoms and also of intestinal lung disease. So that's very promising, but for pediatrics, there's a lack of pharma-sponsored studies at this moment. So we really need academic sponsored trials. And since Kymriah, there's no registered indication, so we still have the one product available. So there's need, and there's also room for point-of-care production in academic collaboration, but numbers are very limited, so we need to collaborate. So A is the normal workflow with the industry. Phase 1 studies are often done in a single center fashion, but thereafter we need to expand, we need multiple sites, we need products to travel, not patients to travel. So that's what we're working on, and the direct call will help establishing this for pediatrics in the near future. We have a lot of collaborators within Europe, in the United and the European Pediatric CAR T-cell Task Force, a lot of collaborators in the Influence, and of course, a lot of people working in the Maxima on CARs. And thanks again for the opportunity to present our data, and happy to answer questions.
Thank you. Thank you very much. We have time for one question, probably. If you have any.
I have one question. I was wondering, are the concerns about the long-term effects any different between children versus adults, or?
No, that's a great question, and we are studying that at the moment. And I think the major concern 2.5 years ago was the vector integration in benign cells, mainly benign T cells, and leading to T-cell lymphoma or TALL. There are several cases that are linked to this, but only not even a handful where it's been shown to be a causal relation. We look back in an international collaboration where we gave input, and in over 3,500 patients with over 5 years follow-up, there was no single case of TALL or TLBL resulting from vector copy number integration. So secondary malignancies due to CAR integrations, that's not a major concern, as chemotherapy has a higher risk, and especially TBI, total body irradiation, with stem cell transplantation, has a much higher risk of secondary malignancies. But what is a concern is the persistence of CARs. So we need at least 6 months, but we probably don't need 5 or 10 years of the CAR persistence leading to B-cell aplasia and agammaglobulinemia. So we tend to give all patients IVIG, independent of complaints, but despite suppletion of intravenous or subcutaneous immunoglobulins, we do see, or we tend to see, I have to say, more infectious complications long-term, like chronic bronchitis. And we're investigating this at the moment. And that could be one of the concerns when we move CARs forward even further. I think replacing transplant is of utmost importance, but often people ask, should you just replace all chemotherapy? And not yet, I would say. Hope that answers your question.
Yeah, thank you very much. We have one quick question from the audience.
Yeah, my name is Foggewitt from St. Anna Children's Hospital, and greetings from Lizzie to you and your team. She was till last year in your hospital working. And I have a question. We calculated the number of facilities we need for phase 4 trials when CAR T-cell will enter the BFM studies a little bit more in the first line treatment. So it's a huge amount of facility space you would need. And my question is, the academic model is a nice model, but it's a very diverse model that needs many facilities. Isn't it a good idea to found academic-driven facilities, non-industrial facilities like Red Cross or transfusion medicine with higher capacities to run 4 or 5 productions at a time, or even 10 or 20 at a time, because every institution needs to go to the legal procedure. It goes to the authorities that need many time, many resources, many money to be certified to make the production in different centers. And I know that Netherlands is very centralized, which is very good. So what's your opinion on that? Would you have more a big centralized facility or a diverse?
That's a great question. I think we can discuss this for hours, but I don't have the answer, I would say. I think for the early phase trials, you need decentralized production, and at some point we need the product to travel. So we don't need a production center in every country, for example, but we need a few to allow for access. And but for phase 3 and phase 4, or to replace any chemo, then we will need more. But we're unfortunately not there yet. And I definitely don't want to replace pharma. I want to be the forefront of pharma, and they should take over when numbers are too high. And maybe Sanquin will take over in the Netherlands at some point, I hope.
Okay, so thank you very much for the three speakers, and we will have a rest now, and we will be back at 4 o'clock. Okay.
I'm all about that bass. I'm all about that bass.
You said you loved me, never said goodbye, but I know now that just was a lie. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. Yeah, you said you loved me, gave me stars above. I thought, sure, baby, I had all your love. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. You used to walk and talk, darling, jump and sing, but I know now you don't mean a thing. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. I'm just a mixed up fool. Mixed up all because of you. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. Now you listen here, baby, what I have to say. If you don't want me, I'll be on my way. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. Now you listen here, baby, won't you come on home? I'm sitting here crying and I'm all alone. I'm a little mixed up about you. I'm just a little mixed up and I don't know what to do. There's a sour in the wind blowing down the road I've been. I can hear it cry while shadows steal the sun. But I cannot look back now, come too far to turn around, and there's still a race ahead I must run. I'm only halfway home, I got a journey on where I'll find the things I have lost. I've come a long, long road, still I got miles to go. I've got a wide, wide river to cross. I have stumbled, I have strayed. You can trace the tracks I've made all across the memories my heart recalls. But I'm just a refugee. Won't you say a prayer for me? 'Cause sometimes even the strongest soldier falls. I'm only halfway home, I got a journey on where I find the things I have lost. I've come a long, long road, still I got miles to go. I've got a wide, wide river to cross. I've got a wide, wide river to cross. I've got a wide, wide river to cross.
Dans le Paris des petits quartiers, oubliez les touristes, les cartes postales et leurs vieux clichés. La tour Eiffel la joue aux yeux des passants, mais moi je vous emmène à Ménilmontant, venez prendre un petit verre, vous êtes mes invités. Dans le Paris des gens ordinaires, on est plus souvent sur boulevard Voltaire qu'aux Champs-Élysées. La tour d'argent fait de l'œil aux dignes chalands, mais y a la goutte d'or rouge dîne en marrant autour de la table de la simplicité. Au marché de Belleville, on entend chanter les accents mélangés. Au bus de Saint-Ouen, l'esprit de Django plane sur les bistrots. À Paris sur la même, je vais me ressourcer au milieu des parfums de fleurs d'oranger. C'est à Paris Montmartre que vous m'avez connu, vous allez découvrir les musiques de ma rue. Dans ce Paris, moi j'aime planer, loin des clichés, des cartes postales et des vacanciers. Les beaux quartiers vous sont familiers, mais là je vous emmène, vous découvrirez ces endroits oubliés qui me font tant vibrer. trois maillés, la voix défoncée. Je vais croquer ma femme en col, son comme à la maison. Marcher des enfants rouges m'emmène autour du monde. Au métro Robespierre, tous mes potes vagabondes. C'est à Paris Montmartre que vous m'avez connu. Venez donc écouter les musiques de ma rue. Dans ce Paris, j'aime me balader au gré des jardins, des portes cochères et des escaliers. Tous ces quartiers me sentent familiers, avec des gens simples et de la sincérité. C'est le Paris que j'aime partager. Ah, c'est déchirant. Et si on essayait... Aïe. Ah, oh, les mecs, et si on... Arrête. Si on essayait une version big band avec des cuivres, trompettes, saxophones et tout ça, je pense que ça pourrait être vachement bien. Aïe.
You're impossibly lovely.
You're out of what you said.
Oh, you do fascinate me.
You're smooth as hell.
Oh, I'd love to get closer.
You really do have your lines down.
You're a cool one.
Wonderful song that's playing.
Would you like to dance?
I didn't plan on staying.
Stay and take a chance.
You came out of nowhere. You're taking my breath away.
May I make a suggestion?
I'm afraid to hear.
Would you like to get closer?
You're awfully near. I'm inclined to believe that you're flirting, but I kinda like it.
Making progress.
Wonderful song that's playing.
Won't you say you'll dance?
I can't believe I'm staying.
We only get one chance.
You came out of nowhere.
You're taking my breath away. Cause you got the something.
What's that something?
Just the some drives a man insane.
You got that something too.
That's something I can't explain. It's just you got the something.
What's that something?
Just the some drives a man insane.
You got that something too.
That's something I can't explain. Things we regret the most are the things we didn't do. You're impossibly lovely.
You're out of what you
Oh,
said.
you do fascinate me.
You're smooth as hell.
Oh, I'd love to get closer.
You really do have your lines down.
Yeah, you're a cool one. Wonderful song that's playing.
Should I take a chance?
I know you didn't plan on staying.
I'd have loved to
You
dance.
came out of nowhere. You're taking my breath away. Cause you got the something.
What's that something?
Oh, the some drives a man insane.
You got that something too.
That's something I can't explain.
I don't need to explain.
Who needs to explain? Oh, that some drives my heart insane.
You got that something too.
There's something I can't explain. Who could have ever imagined there'd be me and you? But they say that the things we regret the most are the things we didn't do.
place that I know where the sycamores grow and daffodils have their fun with the cares of the day seem to slowly fade away in the glow of the evening sun. Peace when the day is done. If I go there real late, let my mind meditate on everything to be done. If I search deep inside, let my conscience be my guide, then the answers are sure to come. Don't have to worry none. When you find peace of mind, leave your worries behind. Don't say that it can't be done. With a new point of view, life's true meaning comes to you, and the freedom you seek is won. Peace is for everyone. Peace is for everyone. Peace is for everyone.
I never cared much for moonlight skies. I never went back at fireflies. But now that the stars are in your eyes, I'm beginning to the light. Used to ramble through the park, shadow boxing in the dark. Then you came and caused a spark that's a world on fire now. I never made love by lantern shine. I never saw rainbows in my wine. But now that your lips are burning mine, I'm beginning to see the light. Used to ramble through the park, shadow boxing in the dark. Then you came and caused a spark that's a world on fire now. I never made love by lantern shine. I never saw rainbows in my wine. But now that your lips are burning mine, I'm beginning to see the light.
play begin. We're actors, and here's our game. Life's a stage where it takes your best to win. So give it your all, folks, while you may. Too soon the shadows may fall, and someday that curtain will fall.
If you are rich, if you are poor, it's all the same, I'm sure. When the curtain comes down, just go along, doing your best, and you'll find peace and rest. When the curtain comes down, what if everything goes wrong? What if sorrow comes along? Hide your feelings from the throng. Life can't always be a song. While you are here, if you play fair, you will be welcome
stepped down from my vice presidency because of being elected for Esfh, to have this part with the Japanese, to be in trouble, you know, with that position. So when I said, okay, I will concentrate on Esfh, and then I tried to bring them, and then we had the idea to have a joint Esfh Conference. Esfh is absolutely nothing. Absolutely nothing. We invited him in 2023 to Berlin. We made a deal to say, okay, you can have a workshop, you know, for Esfh, you can have a workshop here. The next time we can come to you as an Esfh or an Esfh workshop, but they never. And then we discussed it with them several times, and they said, no, this is our office is not interested, and it's a question of money. And they said, no, we can take care for our own speakers. So there's just free registration, no more. No. So what's the problem? For us, you have to pay everything. So I'm speaking next week in Esfh meeting. Yeah. I have to pay everything for this. I only had 3 nights in the hotel. I have to pay my flight costs. I have to pay my registration, which is $800. Yeah, that's ridiculous. Yeah, that's ridiculous. 800. Yeah. What do you get back from it? Yeah, that's ridiculous.
So, welcome all of you. My name is Hans Vrielink. I'm past president of ESFH and immediate past president of WAA. And together here with my colleague, Dr. Wolfgang Ramlow, we are sharing the session. And for me, it's a little bit special, because this is the AAA session, and the AAA session, nor Dr. Ramlov nor me are involved in the AAA session, so I had to look first, what is the AAA in fact? The only AAA I know is the Anonymous Alcoholic Association, but I learned very quickly from my friend and colleague Volker Witt from Austria, from Vienna, that this is the Austrian Apheresis Association. So welcome on this specific session, and I give the word to my colleague.
Yeah, also a warm welcome from my side, and you again, yeah. You are Dutch, I'm German, so we represent the AAA. And Volker and Kurt, we have been friends for a long time. And you are accomplices with Gerda. She's obviously not here. Yeah, da. Oh, sorry, Gerda. So I missed you, and you put us in this position, and you gave us the title. So we are curious. Of course, it's a privilege to represent the Austrian Apheresis Association. And because of our friendship, I think we are qualified to do this. We feel like Austrian. So Kurt Defler, again, has been a good friend of mine for many years. And I'm actually an East German. And after the wall came down, we had a lot of you actually were the first where we exchanged our experience in immunoadsorption. So that's why you established the largest apheresis center in the world at General Vienna Hospital with a lot of experience. And Volker, you are the guy with the pediatric patients having all the experience. And Gerda we met as well with the site apheresis, site adsorption at Vienna. So yeah, and now we are curious, Volker, what you're going to talk us about lipid apheresis, and maybe you will also address the issue of the future with regard to the newly developed drugs. Please, Volker. Kurt, sorry. Volker is there.
Thank you for your kind invitation to this Congress to talk about LDL apheresis. And there you can see that familial hypercholesterolemia is still underdiagnosed and undertreated in general population. And if you look at this data from Austria, there you can see that in patients who experienced their first myocardial infarction before the age of 35 years, in less than 5%, a familial screening was performed for FH. And this is the reality in Austria and in many countries of the world. When you look at this there, you can see what is the importance of long-term exposure to elevated LDL. And there you can see that in patients with the homozygous familial hypercholesterolemia, the mortality rate in patients is about 80% die before the age of 18 years. And even in untreated heterozygous hypercholesterolemia, there's an excess mortality rate in these patients. And there you can see that most of the patients experience the first major cardiovascular event before the age of 40 years. And there you can see what is expected due to the possibility to really lower LDL cholesterol lower than 50, there might be a tremendous gain of life years for these patients. And there you can see what is the reality in the world. There you can see in most of the countries, hypercholesterolemia is not detected, is not detected, and in all patients where hypercholesterolemia is unknown, all these patients are untreated. And there you can see that's what is happening in the world, and this has been published in 2020 in the Czech journal. There you can see what is the expression of homozygous hypercholesterolemia. In Austria, one of 250 inhabitants suffers from this disease, and there you can see some patients present these serious xanthomas and in the tendon, in the cutaneous, and xanthelasmas. But more important is the development of very early cardiovascular disease. And there you can see a pathohistological staining of a boy who died due to acute myocardial infarction at the age of 4.5 years. And there you can see what is in this staining. There you can see there's a tremendous accumulation of lipids in the arterial wall. And this was complicated by this thromboembolic situation. And this is the left coronary artery vessel. And there you can see that this problem developed from the age of birth to the, within 4 years, we had this tremendous development of atherosclerosis in the cardiovascular vessels. First successful long-term extracorporeal treatments were presented by Thompson, Miller, and Breslov in the year 1985. And he found siblings who were both concerned by homozygous familial hypercholesterolemia. And he decided by lot to treat one of them by long-term and frequent plasma exchange. And there you can see there was a tremendous gain of life in the patients who were treated by plasma exchange. And this was the reason that within a few years, more selective devices were developed to more specific reduce circulating ApoB-containing lipoproteins. And this is a patient from the General Hospital in Austria. There you can see this was a system depending on an initial plasma separation, and there you have reusable columns, and these columns, the mode of action was that there were sheep antibodies directed against ApoB. And these columns could be used up to 80 times. Yes, more than 1 year. And there you can see we performed in this patient suffering from homozygous hypercholesterolemia a vein-to-vein approach. And he performed, he's now on treatment for about 20 years, and still we use the same veins in these patients. There you can see now we have 4 different systems to remove atherogenic lipoproteins from the circulation. And the most important are the adsorption columns, depending on dextran sulfate, covalently linked to cellulose beds, or polyacryl-coated polyacrylamide pads. And there you can see the HELP system, the heparin-induced LDL precipitation now has less important, whereas the double filtration plasmapheresis also less effective in removing atherogenic lipoproteins has gained more and more importance, in particular in Germany. There you can see what is happening during a single apheresis. There you can see by a single apheresis we can remove about 70% of LDL cholesterol and also Lp(a). And there can you see within 1 week about 80%, 85% are resynthesized by the liver of these concerned patients. And you can see this is an absolutely safe procedures. Only in 2% we see serious side effects, mainly due to bradykinin reactions. But side effects of any kind were reported in about 2.1%, and most of these complications were due to the use of 16 or 18 needles, and the most common complication is due to the needle insertion to native veins. Other rare complications are hypotension. This is explained by the reason that we need a large blood volume in the extracorporeal circuit of about 400 milliliters. And therefore, we don't treat children with a body weight less than 15 kilograms. But rather rare side effects were headache and hypocalcemia due to citrate anticoagulation. And when you perform apheresis for a long term, you can reduce the pre-treatment concentrations of LDL and Lp(a), and this might be one of the reasons that LDL apheresis is effective. And there you can see a publication in the atherosclerosis, thrombosis, vascular biology, and this is the statement from the American Heart Association. And there you can see the FDA proves the indications of homozygous familial hypercholesterolemia, but also patients with progressive atherosclerotic vascular disease and elevated LDL concentrations greater than 100, or patients with an Lp(a) greater than 60 milligrams should be treated or can be treated by LDL apheresis. And there you can see this is the well-known reduction rates of LDL and Lp(a). And there you can see that the reduction in major adverse cardiovascular events in this review article was 75 to 95%. Yes, so most of these patients become absolutely stable for their coronary artery disease. And to my personal opinion, it is rather important that the myocardial microvascular perfusion is improved during long-term LDL apheresis. And there you can see in difference to conventional drug treatment, LDL apheresis is able to change biomarkers for endothelial function, coagulation, and thrombosis, and rather important, the oxidized LDL and other inflammation and oxidation markers are seriously decreased by apheresis. And this might be responsible for these high rates of stable patients under frequent LDL apheresis. This is a publication by Hokiruka, presented by Mapuke, and he demonstrated that the rate of progressive disease and the occurrence of major cardiovascular events under the maximum of medical drug treatment compared to patients who in addition received LDL apheresis. And there you can see that there's a 70% reduction in the risk of a very serious cardiovascular event. And when you look at this, we still are waiting for any drug that's able to reduce the rate of MACE by about 70%. When you look at the PCSK9 inhibitors, we have a reduction rate of 12%. We should consider this if we decide to treat patients with LDL apheresis. There you can see this is evolocumab, yeah. It's not a comparable rate of reduction in MACE as to the other one. For homozygous patients, we have two new drugs. This is the lomidapib and the evinacumab, and both substances are able to reduce LDL cholesterol, not via the LDL receptor. And the reduction rates of these both medications is about 45 to 50%. Yeah, and none of these substances could demonstrate any beneficial cardiovascular effect. Yeah, and there you can see the price of these two substances, and this might explain why these both drugs are not used in a great number of patients. When we got the PCSK9 inhibitors, we had a marked decrease in patients with the heterozygous hypercholesterolemia treated with LDL apheresis. But at this time, we started to treat patients with elevated Lp(a). And you know, Lp(a) was detected by Kari Berg in 1963, and Lp(a) is an ApoB-containing atherogenic lipoprotein associated with increased risk of atherosclerosis, vascular disease, stroke, and very important, of calcific aortic valve stenosis. And when you see what is the occurrence in the general population, there you can see that about 50 to 20% in the general population have elevated Lp(a) values, and about 2 to 3% have very, very high values, greater than 150 milligram per deciliter. And Beate Jäger was the first who demonstrated that in these patients, lipoprotein apheresis was able to reduce the occurrence of any MACE, as a major adverse coronary event, by 86% when she compared these patients to their rate of MACE before apheresis treatment. And this was published in the Nature, and the results were assured by a publication by Lebmann in 2013. When you look at the most important publications, as to my personal opinion, you have to look at the German lipoprotein apheresis registry. And there you can see that Franz Heigl in 2015 compared 4 different types of lipid apheresis methods in 118 patients with coronary artery disease and elevated LDL and or elevated Lp(a). And during a mean duration of 6.8 years of apheresis, a reduction in MACE of about 80% was reported when compared with a mean period of 6.4 years before apheresis. And the German lipid apheresis registry is a large multi-center prospective investigations in 2,028 patients with elevated LDL cholesterol or Lp(a) levels or both. Comparison of MACE and non-coronary events during 5 years of LA with an interval of 1 to 2 years before LA. The authors could detect such reduction rate in MACE of 74% and non-coronary atherosclerotic cardiovascular events of stroke, carotid artery surgery, and new peripheral vascular events of 66%. This is an absolutely important publication presented by Volker Scheckler. When you look at the guidelines to treat the patients, you can see that you have the recommendations from the FDA, you have the recommendations for the UK Heart Association and the German and Austrian Association, and commonly all these guidelines have similar indications for long-term lipidapheresis. That is the homozygous lipoproteinapheresis, the heterozygous patients with insufficient response to conventional treatment, and an increasing number of patients suffering from elevated Lp(a). The lipidapheresis is absolutely safe and efficient procedure and has only few disadvantages. That the procedure is rather costly and requires high patients' compliance. When you look what is the pathway to organize a lipid apheresis unit, there you can see the recommendations from Barhofer and Waldmann, and they both recommended apheresis for patients suffering from atherosclerotic vascular disease, either with elevated LDL cholesterol or lipoprotein A greater than 60 milligram per deciliter. And there you can see we have to optimize all other risk factors. We have to institute the maximal drug therapy, and if the LDL goal is not achieved or a worsening of the vascular disease due to elevated LPKNA is present, we should consider lipid apheresis in these patients. I come to my take-home message. There you can see, I just already told you that lipoprotein apheresis is safe and highly efficient and should be performed in particular in homozygous patients for primary prevention. We shouldn't wait until any cardiovascular complications occurs in such patients. We have to start in these patients prior to the age of 6 years in most of these patients. And we should see what is happening with Lp(a) in the future. Thank you for the audience.
Yeah, thank you very, very much for this nice and comprehensive overview. Are there questions from the floor? Maybe I have the first question. Do you have an idea about the number of lipid apheresis patients in Austria? An idea?
The Institute Athos, where I now have 80 patients in the General Hospital, 31 patients, and in whole Austria about 140 patients.
Okay, in Germany, we do have about 5,000 patients on continuous chronic lipid apheresis treatment. Meanwhile, about 50-50 LDL and Lp(a) or combined LDL and Lp(a). What about Spain? You mentioned it. 200. Yeah, you mentioned 200 treatments any other week. Would mean about 10 patients at your clinic. All homozygous or? Heterozygous. Okay. What about Czech Republic? What about?
We have 6 patients, 5 homozygous and 1 heterozygous.
Okay. Cool. And all over the country? Yeah, we offered several also other people who are interested. The German registry, we even translated it into English, but it is not a big deal to adjust it to other languages as well. So we could even expand our database. So it's just an offer. We discussed it earlier several times. So, and I know about activities in Spain, UK. Yeah, also UK is also here. UK, I saw someone from UK. Is there anyone having an idea about? Okay, but it's your talk. Sorry. So
I was wondering, what is the difference between Germany with 5,000 patients and Austria with, when I calculate fast, about 200 patients in whole Austria? So I can't imagine that Germany is 20, 30 times larger than Austria. So what is the difference? Is this what you said, the unknown testing or the unknown patient, or is it in another way, because what I'm familiar of, we have only 5 families in the Netherlands, and it's only treated in one center in the Netherlands, so it's very limited.
It's very limited. Also, UK had about 60 patients several years ago. France was 200, but the numbers went down. Italy also about 160. That's what I know. So it's totally different, but I think it's the availability or the accessibility to the products. So most of the technologies, the selective or specific technologies, are from Germany. So that's why, okay, there is marketing, there are marketing activities, but on the other hand, B. Braun, the HELP system, was the first system, and it was actually launched to the market before the first statin came out. At that time, in 1986. So that's why, and then that was a temporary approval at that time, but it continued over the time. So it is the same as with steroids or with aspirin. There are not major trials you can cite because it's already established. And I think that was the reason that in Germany it had been established very early. The systems were available, and then we had the rules, the specific rules for that.
No, I generally agree. Germany is the most prominent country for lipidapheresis. And even in the United States, you'd have a distance for the next center that provides lipidapheresis of about 500 miles.
Yes.
Yeah? And in Austria, we don't have enough centers that perform lipidapheresis because the reimbursement at the hospital is too low. Yeah. And this is the main complications, and we only have in Austria one private institution, that's the Institute Athos, that's performing lipidapheresis and get sufficient reimbursement.
And I want to add, because I think Germany has not a program against to find Lp(a) and LDL apheresis, but in pediatrics in Germany, the organization is much more stronger. So there is the recommendation when until the age of 16, every patient in a pediatric department should have an LP(a) examination, and one is for the life enough. And so I can tell you, in my hospital, we have also this program, but you know, maybe there is this controls in childhood from newborn to the age of 5 and now to 12. It's not in this prophylactic program, and I think that's one thing we have to care about, because it's ridiculous that in most of the countries, 90% of the patients are not detected. Otherwise, one of the family members is died, and that's a pity.
I completely agree, and also I want to support your mention. In Germany, according to a survey of Ulrich Julius, we have more than 300 apheresis centers. So that means for any patient, there is an apheresis center within the reach of 30 minutes, even by public transport. So I think that's one.
In the Netherlands also is screened, and the youngest one we did was 3 days of age. Of course, not lipid absorption, but a TPE, because 400 mils is a little bit much for a child of 3 days of age. Any questions? Any other questions? I think then, thank you so much, Professor Dervla. I have the honor to announce my co-chair now for the next presentation. We just during break had a discussion on soccer. Yeah. There's always a war going on in soccer between Germany and the Netherlands, and he is in favor of the Netherlands. I can't understand why, by the way, but maybe this is because of his professional background as soccer player in many years ago. And Nowadays, Dr. Wolfgang Remloff is having a medical expertise in apheresis. He is the director and the medical head of the apheresis center in Rostock, in eastern part of Germany, and specialized in extracorporeal treatment, especially LDL apheresis, and then you see the discussions going on between two LDL apheresis specialists and immunoadsorption. So Dr. Derfler is also an expert in immune adsorption, so I think we have two experts in the field here standing. So,
Thank you.
for you.
Thank you very much for this kind introduction. And again, these two friends actually gave me this talk. And I must confess that my clinical experience with immunoadsorption dates back 20 years. So I have no practical skills anymore. I'm involved in organizing conferences. Of course, I'm sometimes involved in organizing clinical studies or criticizing, but they gave me the talk. And you might have recognized I have a little bit knowledge about lipid apheresis. This is my special field. So, but now, unfortunately, I took the challenge because Kurt Derfler said, Gerda Leitner wants you. And I said, no, is this true? Yeah, he said. He said, Gerda wants you. I said, no, I cannot give this talk. So that I took the challenge, actually. So, but then I started searching the literature, in more detail. Of course, I know that the general picture, I feel very well. And 25, 30 years back, I felt we were number 2 in the world, just behind you. So you were number 1, and I felt we in Rostock were number 2. But this is really a long time ago. So I checked the literature, and then I really got confused. The more I read, the more confusing it was to me. So now I try to bring it down, because I could remember I stopped giving, when I was actually the expert in that field, together with him, I stopped giving talks on immunoadsorption overviews 15 years ago, because it became too complicated at that time. And now I learned nothing had changed. So here are my disclosures. First, it's very important to talk or to clarify what you're talking about. Immunoadsorption, we also have one technology in lipid apheresis, where it's an antigen antibody binding by the company of TerraSorb. Originally, they used cheap antibodies. So this is based also on a immunologic interaction. But today, we look at immunoadsorption just as a treatment that is based on apheresis technology that uses adsorptive binding forces to remove immune-relevant cells and or factors. I will concentrate on the factors today. And that's what I said. Yeah, when I checked all the literature, and then I had to find an idea what indications you should talk about. So I decided at the end maybe to talk on emergent new indications in the field you might not be familiar with yet. So, but still, this is the problem. It's rather, so immunoadsorption and apheresis in general, apheresis therapy, is considered as last resort or emergency treatment options. Usually, we have to deal with small patient numbers, and actually, we have a very heterogeneous patient population, which is very, very difficult to have a harmonized treatment protocol, also for clinical trials. That's very, very difficult, and there is no understanding of our subspecialty experts from the other fields, neurologists, cardiologists, so they don't understand, because they are used to clinical trials including 6,000, 7,000 patients, and then you look at the statistical significance, but that is not possible here in apheresis therapy. And there are often even no established protocols, and even no clinical outcome assessments. In particular, you will see later we use clinical or scoring or questionnaires, and then in particular neurological conditions, they are even different, even from the publications we check. So, and yeah, mostly it's not that easy. There is no gold standard to compare with. I actually have been fighting against sham apheresis because sham is passing the blood, you know, from photopheresis if the blood sees sunlight, and in artificial service there is complement activation. So if you talk about immunoabsorption, you have signals going back to the body, inducing any kind of immunostimulation or immunomodulation. So that's why sham is not a placebo. That's very important to learn because most of the clinical trials include a control group with sham apheresis. To me, this is totally stupid because sham has an active action. Yeah, and then also there's another issue in chronic conditions because this is the standard and it's very, as you said, very costly, the procedure. So we are used to have 5 to 7 treatments within 2 weeks, 10 days to 2 weeks. And in a chronic condition, this is not enough. We have to have a maintenance schedule with predefined conditions. Yeah, and then there's another issue. The neurologists do have the patients and the transfusion doctors, and the nephrologists do have the staff and the equipment. And they need to collaborate, and others don't trust us. Or they feel we have even the higher earnings, so that's why should we present our patients to the nephrologist or transfusion doctors here. Yeah, and then if the study takes too long because of the small patient numbers, we have, meanwhile, we have a lot of approval of new drugs. You will see that happens all the time. So if it takes too long, of course, there's no standard of care anymore you compare with in the control group. So you cannot finalize your study as originally planned. And this is the best example. And it's so funny. If you look here, rapid communications, they're rapid. We have been waiting 10 years for the results. 10 years. 171 patients, 1-to-1 randomization. And we criticized the study from the beginning, that this is poorly designed study protocol. And now we have the results. No benefit for apheresis. So obviously, everything you could make worse, it happened here in this trial. And it took really 10 years. And the results were presented about 2 weeks ago when I was in the audience, and they said they will have further subgroup analysis. Why doing subgroup analysis? Because this is really absolutely nothing, but also it reflects our situation. So first, now technical part, you see here the beads usually for adsorption, the beads where the targets are adsorbed here. And you can see here there are spacers, and the spacers are important. Also, if you have longer spacers, you can allow larger molecules to bind. On the ligands, here the green ones here, or green-blue ones, they are important for the selectivity or specificity of binding. And so therefore we do have semi-selective, selective or specific binding. You can influence this here by the ligands. And we have high removal rates for circulating immune factors. We have minimal loss of essential factors in pharmaceuticals compared to plasma exchange in particular, and no replacement of proteins or cells is required. This is just to give you an overview. If someone is interested, I can send you a PDF. If someone is still interested after my talk, I must say, I can give you the presentation as well. So just as an idea, we do have a lot of ligands to remove a lot of immune factors here. And these are the principal application modes. And you know that in lipid apheresis, as you talked about, we do have systems where we can use the adsorption cartridge here in the whole blood system, but there is no, I specifically search for that. If meanwhile, if there were some developments, but here the adsorbers are used in the plasma circuit. So I don't know any adsorber to be used in the hemoperfusion circuit for immunoadsorption. And then because if you do have one single adsorption, then that's the problem with saturation. So for instance, we have the tryptophan 350 column, and this column can only be treated by or passed by 2.5 liters of plasma. Then the column is saturated and desorption takes place. So this is, it's a difference, and that's why, because of the saturation process, some engineers had the idea we can regenerate the columns during the procedure. So that means you use 2 columns, and while one column is being passed by the plasma, the other column is being regenerated. So you can alternately use these columns here. You can see here in the middle here, the 2 columns here. So you have almost unlimited capacity by using the system. So because you always have a fresh column during the procedure, and in most of the systems, the columns can be reused in the same patient. Up to 5 times is the regular, but we use it up to 20 times. And then there's another system, we will come back to this, with only 1 column, where they use the 1 column in between, so the plasma goes in bypass mode, and the column is being regenerated, in particular for the Pentrasorb system, and I think for the Securim system as well. Yeah, so the regenerable columns allow us to have unlimited capacity. So I can remember patients with rheumatoid arthritis 20 years back, where we treated about 14 liters, and it took up to 8 hours of treatment at that time. Yeah, Volker, you're familiar with all this. Yeah, so we can use large treatment volumes, and we don't need substitution again. But because of the switching and the rinsing procedures, you have an unspecific loss of some plasma. That's about 10%. So there's also reduction of other. It's not specific, although the binding might be specific. There is some loss of other factors. Here, just showing you the TR350 is a single column, and you see the reduction rates here, circulating immune complexes, and this is a patient with patients with myasthenia gravis, up to 55%. To my clinical experiences, rather 30 to 40% during one procedure is not 55, because the Japanese are very small. This is based on Japanese data here as well. And you see this column has to be used several times in order to bring down the autoantibody here, see before immunoadsorption. But by using it 5 to 7 times within 10 days, also you can really achieve a low autoantibody level. Again, this is the double column system here. The double column system with unlimited, and there's just one example for time reasons here, and this was as early from 2000 here, this publication. You can see you can reduce the IgG level by 80%, and this is true as well for the autoantibodies. So during one procedure, up 85%, yeah? That's true. So, and after 5 treatments, it was the schedule they used for dilated cardiomyopathy from Monday to Friday. I think it's not very clever, because you need some time in between to allow redistribution of immunoglobulins from the tissue into the blood circulation. So I think it was not the most clever strategy here, but it shows you it goes down from 11 gram per liter here IgG to 0.8. And this is, yeah, you know, we started at that time because we were a little bit scared of infections. We started initially by supplying IgG at the end, 10 gram, and then we realized we had a lot of phone calls at that time, and then we realized there is no need to substitute IgG even in patients with infection risk. Yeah, summary of technical part. We do have different columns here. Reduction rates vary from 35 to 85, yeah, up to 8 hours treatment with the double column, regenerable columns. So excellent safety, not discussed here, and we do have specific ligands to remove a wide spectrum of autoantibodies. So I think this is the first indication. I think you're much more familiar than me with this indication. I think there's no doubt that immunoadsorption or plasma exchange has a general role in desensitization of kidney transplant recipients, also in solid organ transplantation. You said at General Vienna Hospital for lung transplantation, or as well as an antibody-mediated rejection here. I think this is very clear. I just want to focus or to emphasize this one publication from, Georg Böhmich is from your group originally. He continued his work, and this is a summary from an expert consensus. Here, a group from all over Europe from last year, and NDT published, and these are all the antibody depleting, complement inhibiting, or other immune modulating agents. So the statement of the authors was, there are no established protocols in all the different groups. They actually called for joining for clinical trials together with the new novel drug developments. But on the other hand, they said building on the 2019 expert consensus, we recommend apheresis, either plasmapheresis or immunoadsorption, as a mainstay in the treatment of early antibody-mediated rejections, primarily caused by preformed donor-specific autoantibodies. And this is a nice graph to show here. So if you want to remove the autoantibody or the antibodies very fast, then immunoadsorption is a very, very good choice. And for me also, I would, in this case, as the author suggested, although there is no evidence by the data, but they suggest that immunoadsorption might be the preferred method here. Yeah. And they also suggested a treatment scheme here, how to proceed with those patients. And I want to bring to your attention another publication I found from India, where the religious background is a little bit different. So they have no cadaver kidney transplantation, so they rely on living donor transplantation. And this is a group now summarizing from New Delhi, summarizing the results of 21 patients here with ABO incompatible blood group transplantation here. And you can see this is a standard procedure. They use rituximab. Together, not plasmapheresis, they used immunoadsorption with the Securim device, which is a device that has oligosaccharides as the ligand that specifically binds anti-A or anti-B antibodies here. So they used 3 treatments together with rituximab, and then you can see their goal was to have the titer isoagglutinin titer below 1 to 8, and you see they achieved this in all the patients. And here you can see wonderful results. These are the creatinine levels at discharge from the hospital. That's quite amazing. 21 patients. There's another series from Canada and from other groups as well, but I think this is established. Now, to here, it was very simple. I think everybody can follow this, but now it's getting complicated. We talk about long COVID or chronic fatigue syndrome as one of the lead symptoms, you see. So about any month, there is a new publication on autoimmunity and detection of many new autoantibodies in long COVID. So and we have to change our view on autoantibodies because obviously there are so many autoantibodies that have a regulatory role. And what we had to learn as well is that G-protein-coupled receptor autoantibodies, they are rather agonistic antibodies. So they activate the G-protein receptor or G-protein coupled receptor antibodies. They are in the membrane of the cells, and they do have the responsibility to transfer the signal from the blood, from the outside, from the cell into the cytosol. That's there, and we have more than 1,000 of such protein receptors. And on the other hand, you see the great variety of symptoms here in those patients. And again, there's a lot of publications. I could easily add more about autoimmunity background, at least in a substantial group of long COVID patients. And in addition, 2 groups from independently, one from Amsterdam and the other from the Yale University, were able to show by transferring purified IgG from patients, healthy volunteers, and patients who recovered after COVID into mice. They could really transfer symptoms, COVID-like symptoms. Of course, mice are a little bit different from the humans. So, but they could transfer by injecting the IgG into the mice. And it was independent, so it's another proof of that immunology plays a substantial role here. Yeah, here, this is a nice publication showing that there is some overlap from the symptoms from post-COVID and chronic fatigue syndrome. And this is very important because chronic fatigue syndrome is the lead symptom of long COVID patients. That's important because we have clinical trials. And here is a review on 57 studies comprising more than 250 survivors after COVID-19. And they said the most sequelae included mental health, pulmonary and neurologic disorders. And this is very interesting. And also, this is data from Germany. That's a mathematic model showing this is the long COVID. These are the long COVID cases, and this is the prevalence here, the yellow line, the broken line. This is the prevalence of chronic fatigue syndrome. So I think there are some parallels that it goes up. Usually chronic fatigue syndrome has been known after EBV infection and other viral infections, so mainly EBV, Epstein-Barr virus. And to give you an impression, because I had no idea about this for chronic fatigue. I'm also a little bit tired after work, but this is not like tired. I want to give you can read on your side on yourself. You see, this is from email correspondence with the patients. Since the coronavirus infection, I have unfortunately been reduced to a complete wreck and feel as though I were 104 years old. Another patient, since my COVID-19 illness, my life has been thrown completely off track. At present, I'm unable to continue my work as a teacher. Managing everyday tasks is not possible without help. Even caring for the children can only be managed with the support of others. And then here another female, my walking distance was approximately 30 meters and I could barely hold a head of cabbage. These were all patients initially healthy before they got vaccination or coronavirus infection. They were all healthy, they were active, they actually were active in doing sports, and these are the parents of a 13-year-old female here. By now, her fatigue syndrome has become so severe that she can no longer even sit upright. She is also so weak that she cannot hold up her head and is unable to hold on when being carried. Though we have a lot of young females in particular, they really can't go to school, they can't study anymore, and they were really healthy before. It's not psychosomatic, of course, as a consequence in this condition. I think that's very clear that you have some problems with your life. So, and the major sentence of many of the patients is, I just want to have my old life back. That's what I would like to have. And we do have Carmen Stein from Charité already started doing immunoadsorption already in 2016, so before COVID, on Epstein-Barr virus patients. In 2016, they did the first immunoadsorption studies, and this is their latest trial here, where they performed immunoadsorption in patients better to adrenergic receptor autoantibodies, because they, during their work in the last 8 years, detect or found a correlation of beta-2, beta-1 adrenoceptor antibodies and muscarinic receptor. So they are measuring beta-1, beta-2, M3 and M4 antibodies. This is IgG, IgA, and IgM after or during 5 immunoadsorption sessions within 10 days. That's what they did now. And there's a paper on Lancet, and I think we can really celebrate to have an immunoadsorption trial in Lancet. It's really amazing here. And they could prove on the long term. And the clinical results here are 14 out of 20 patients responded according to the SF-36. This is a questionnaire and the Bell score. There are several scores. I tried for a registry to put together the scores. I found more than 20 scores being used to identify those patients. But SF-36 is accepted as well as the Bell score. And the added value of the study, as the author stated, was remarkably these clinical improvements after 5 immunoadsorption treatments were observed as early as 4 weeks post-treatment and lasted up to 6 months. In some patients they did after 2 years another series. And now these are the studies underway now. We have several studies, randomized, controlled, and unfortunately sham controlled. There's not the time to discuss this. We can do this later on. Sham control trials here from Hannover. You see patient numbers are rather small. And this is on double column, this on double column. And here the POCO trial is on the tryptophan, the single column adsorption here. And also Amsterdam has not started yet with the trial, and they are going to use the CR350 as well. So next topic, it's as same as complicated as long COVID, neurological diseases. So, and I used ChatGPT to say, please create a graph and show me all the autoantibodies in neurology and the locations where they bind too. And this was the result. And it took only 2-3 minutes. I was really astonished, yes. And then I said, oh my goodness, you should continue. You have no clue of this topic here, so go on. And then I said, please, could you make me a list of all the other antibodies related to the diseases? And then I got shocked here, and this is really true. During the last decade, so many new autoantibodies have been discovered. But again, it's not like myasthenia gravis. You do have 1 or 2 autoantibodies, and you have 1 epitope for binding, and then it's blocked, it's inhibited. This is a lot, a huge array of autoantibodies. So we have really to change our understanding of this. Yeah, and then I asked, okay, that's too much for me as a simple nephrologist. Please look at the prevalence. So this is all by ChatGPT, and I checked it afterwards. I checked the papers, and so I do have the sources as well. You can see it there. You see? Yeah. You can see it there. There are the sources. And I said, okay, multiple sclerosis, myasthenia gravis, autoimmune encephalitis, and then CIDP. We are familiar with that, and I only can focus on this. There are several papers on that, and again, a lot of papers, case series from China as well, from all over, from India, and of course a lot of from Germany and other European countries. I want to address a few ones. You see, this is here the major indication: CIDP, Guillain-Barré syndrome, myasthenia gravis, multiple sclerosis. And then the neurologists. This is done by the neurologists themselves, so they are motivated when they do it on their own. No need to talk to the nephrologist, to convince the nephrologist to stop dialysis treatment and to do immunoabsorption. But here you see 153 patients, and then they compared usually plasma exchange and immunoabsorption, because plasma exchange was already established in the guidelines. But the comparison is, for instance, plasma exchange, plasma volume 0.7 times, patient's plasma volume, immunoabsorption 2.5 times plasma volume. And immunoabsorption, that's the core message for today. If you go home and have this one message, immunoabsorption is not immunoabsorption. One is 2.5 liters, one is 2.5 times the plasma volumes, totally different. And if you remember this, then it's fine. Then I made a great job then. So, and this is autoimmune encephalitis from the leading group here in Berlin, Charité, Harald Preuss. They discovered most of the antibodies in the last years. And this here, immunoabsorption and apheresis or immunoabsorption is considered first-line treatment here, together with steroids or IVIG. And this is another very important publication that came out a few weeks ago with an impact factor of 10 plus something. Here for CIDP from the group from Ulm, using the scores here, CIDP score and after immunoabsorption. But this is a long-term application. It's not just doing 5 to 7 treatments, but it's on the long term for half a year here. And altogether, this is a summary of 39 patients at the end. So, and I tried to put this together. Neurological diseases here, usually patient with unsatisfactory results to first-line treatment or in clinical or in critical condition. Yeah. So on different diseases, from the papers on neurology, we have more than 500 patients in the summary, but it's no controlled trials, just comparing to plasma exchange, but nothing compared to the original standard of care. And these are here the German guidelines, and I also put the Esfh guidelines, which are very similar, and thanks to you, Volker, and to Reinhard Klingel, also immunoabsorption now is listed among the indication, not only TPE in the Esfh guideline. I think that's very important here that we have. So, and the same is true for the children. Again, 7 years observation period, 39 patients. That's from the German registry, you know this very well. But again, I want to bring over the message. It's a long period of time, it's a small patient number, difficult to run clinical trials, of course. Yeah. Now the last topic, C-reactive protein. It's just, you know it from the lab, yeah? It's very good. C-reactive protein, the goal is not just to show us there is an inflammation, but C-reactive protein has a function in the body. and C-reactive protein is fighting invading bacteria or microorganisms by attacking them here to the phosphocholine binding, and then to the CRP, C1q is binding, and the C1q then initiates the cascade. Yeah. Okay, there are a lot of papers showing this, that because this is not only true to the microorganisms, but also in the body. If the tissue is damaged or necrotic, then also it will be marked by C-reactive protein, and then it will initiate the breakdown. And unfortunately, in myocardial infarction, not only the necrotic material is marked by the CRP, but also the damaged. And the damaged cells actually would have a chance to recover. So that's why it's important. So, and this is the idea of the CRP apheresis in patients with myocardial infarction and elevated C-reactive protein. There is in myocardial infarction bad prognosis with CRP, elevated CRP, and we're waiting for a study from Innsbruck on 202 patients. The results will come out or will be presented in August at the European Cardiology meeting. It's a one-to-one randomization. There's a summary here for my personal idea here. I put it together to say for the future, acute immune-mediated conditions, 5 to 7 treatments, that is fine. And as an emergency treatment or bridging therapy until immune-modulating drugs work, or in chronic immune-mediated conditions, we need a long-term protocol, because there are some trials where it works at the beginning, but then the observation period ends after 1 year. But if you just took for instance, methotrexate for 2 weeks, it will not work and last for 1 year. And the same is true for immunoabsorption as well. Thank you very much. Sorry but it was a complicated issue. Sorry for the time.
I thank you very much for this really interesting presentation. And I can imagine that looking at the number of slides, this is really a challenge.
I brought it down. I got stopped in the elevator today. There's one person, he can prove. Yeah, we were stopped in the elevator. And I said, I need the time to reduce my my slides, the slide number.
Take care of AI. 1 hour ago, I was looking at myself in AI because I was looking at histories and to give some bio on the speakers. I was looking at mine and I saw things that I even didn't know about myself, so be careful of that. Be careful of that. We have time for one burning question.
I have no burning question, but I have a question.
Oh.
But it's not burning.
Oh. It should be short, Professor Leitner?
Yes, thank you. Thank you very much. Really very interesting. I wanted to know if you give the CRP apheresis as a chance, because it was a little. Yes, we wanted to make a study on patients with necrotic pancreatitis, but we didn't do it because of money.
I actually was consultant at that time, and I recommended to the company not to do a study on pancreatitis and also some other indication. I said, why don't you focus on myocardial infarction? Because you do have wonderful results. And the problem, the first trial, we didn't have the time, but the first trial was a control trial, but was not randomized because shortage of staff during the weekend. So that's why during the week, they performed pentrazorb treatment. During the weekend, they did not perform. So this was the control group. So obviously, you could speculate that during the weekend, the care in general was a little bit on a lower quality. So that's why the recommendation was, and the major issue at that time, I can remember, was that the cardiologists were interested, and the cardiologists were confident in the treatment. And so this is from Innsbruck, the trial is an investigator-initiated trial, and there's a randomized control trial, and I think we have to wait for that. There's another issue. The company went bankrupt in between. So now we have a situation in Germany, just last sentence, we have a situation that the German health authorities will fund a study of 500 patients. They made a calculation and said, we will fund an apheresis trial for 500 patients, but at the moment it is not very clear if the system is available anymore or who will jump in into it. That's why it's difficult to answer. Sorry.
Okay, thank you.
But I hope we will have positive results nevertheless.
Yeah, we all can go, I think, and that's very interesting because, and that's a little bit adapting to what I try to say is, we have seen scientists here many years in the field together, I think over 80 years, and so together with me, we're over 120 years in the field working, but we really need young professionals also. So this is really a challenge. We try within ISPT, we try also a little bit here. So it's an honor for me to present now a young scientist, a young professional from the Netherlands. Dr. Oetjes is hematologist in the Leiden University Medical Center and leading the transfusion medicine, so the apheresis department in the Leiden University Hospital in the Netherlands. So it's a pleasure to announce Dr. Claudia Oetjes.
Thank you very much for the nice introduction, and indeed it is a challenge. I'm going to talk about challenges in therapeutic plasma exchange, but it's a challenge to beat 120 years of experience, so I'm not going to do that. And it's also a challenge because I'm the final speaker of today, so I hope to keep you awake until the end. I'm going to talk about the challenges. Where do we stand? I do not have any disclosures, and I want to start with the patient. Imagine you're the physician. It's Friday afternoon, because it's always Friday afternoon. And the neurologist calls you because he has a 32-year-old female, and she has acute neurological deterioration. And he says, well, she was previously completely healthy, nothing wrong since the day of 5. She has a headache, she's confused, and she has behavioral changes. So what do? I'll make it easy. She has seizures, she goes to the intensive care unit, and the questions, and what we see is there are on the MRI scan some abnormalities in the temporal regions. She has a mild pleocytosis, and the autoimmune encephalitis panel is pending. Would you start TPE now? Now, to keep you awake, I want you to join me, and I have 3 options. Who's going to start immediately? No, I want to wait for the results of the antibody screening, or do you start steroids or IVIG first? Let's start with option A. Maybe you can raise your hands or even stand up. Option A. Yeah, we have some in favor of option A. Option B. Everybody can join. And option C. Okay. Yeah. What you see, I think what went on in your mind, should we start, shouldn't we start? Yeah, the antibody results may take days. And early treatment may improve her outcomes. But yeah, I don't want to overtreat this patient. I think this is reality, because often in real life TPE is started before all diagnostic results are in and you have complete certainty. What actually happened was that she was started on high-dose steroids, and after 48 hours she didn't improve and TPE was started. But then there's the second question. Now we're starting TPE. How would you schedule the TPE? Again, 3 options. Would you start with a daily exchange or every other day, depending on clinical response? Who's in favor of option A? Yeah, some in favor. Who would do every other day? A bit more. And who would wait and see what the response is and make the decision based on that? Yeah, also. Yeah, once again, yeah, different ways to go. That's the problem. There is no universal standard how we approach the TPE. It's disease dependent, but also not only ICU logistic matter, but the logistic of your department. If I ask my nurse to do a treatment in the weekend, yeah, she's less available during the week. We have a small team, so it gives new challenges. Protocols may vary, but I think the clinical judgment is key in this. But we start a treatment, every treatment has complications, and during the second session, she complains about perioral tingling, she has some muscle cramps, and her QT is prolonged. Again, what do you do? You stop TPE immediately. You give calcium, you slow the citrate infusion, or all of the above. Who's in favor of option A? Nobody. Option B. Option C. Yeah, option D. Yeah. Sorry? Yeah. I think citrate toxicity is common, and early recognition is critical. But I think it's good to realize that the nurses are the first to detect this. They are the ones that are saying something is wrong, we should do something. The safety of the procedures is a real team responsibility. After 3 sessions, her fibrogen drops, and she has a mild bleeding at the catheter site. Do you change the replacement fluid? You continue, she was started on albumin, so continue on albumin. Are you going to switch to plasm, or are you going to combine? Option A. Who is in favor of option A? Who would keep on albumin? Nobody. Who is going to switch to plasm? Well, I think we're in agreement. Option C. Yeah. Of course, albumin has less of an allergic risk, but gives more bleeding risk because you also deplete the coagulation factors. On the other hand, plasm, you have more coagulation support, but also more reaction. Although with SD plasma we have nowadays in the Netherlands as standard, it's also a less risk of reactions. But every choice you make has trade-offs. Well, after 5 sessions, she slightly improves. She's still confused, and no antibody results yet. Do you continue? Once more. Option A, yes, I started this, I'm going to continue. No, we're going to stop. Or we're going to switch strategy. Who's in favor of option A? In favor of option B? C? it's not a challenge, I think, within the apheresis care. There are no stopping rules, and clinical response, versus uncertainty of stopping too early, what should you do? But on the other hand, if you keep going on, if somebody isn't really improving, you might be doing too much. I think this is one of the biggest challenges, knowing when to stop the treatment or maybe not even starting the treatment. I think what this case gives us, that the TPE decisions are often made without all information. Timing is critical, but also uncertain. There are frequent complications, but mainly manageable complications. What kind of replacement fluid you choose matters, and our stopping rules are unclear. In this case, the patient was later diagnosed with anti-NMDA receptor encephalitis, and she improved after immunotherapy. Did the TPE make the difference? Or would you have improved anyway? Who knows? I want to pick up on clinical challenges, and I think I just named a few. There are a lot of problems, mainly with indication and a lot of evidence gaps. Many indications rely on low quality, low level evidence. If you look at the Esfh guidelines, it's level 2 or 3 category. And there's a significant difference between centers, how they use those guidelines if there's uncertainty. Are we over-treating our patients? Are we under-treating our patients? It's difficult. And what's also difficult, because that's something Hans pointed out, you should realize that the guidelines that are made by the Esfh are mainly on English-based literature. So there's also a lot of evidence not taken into account. It's a challenge if you think about the timing and the treatment strategy. When should TPE be initiated? If you are too early, it might be an unnecessary burden for the patient, but also for the treating staff. Or if you are too late, there may be irreversible damage. And there's the question of interactions with other therapies. Because if you give immunosuppressants and you start your TPE, maybe you lose the effect of the medicine because you lose the medication. And if you want to give IVIG, it's difficult to combine it with TPE. Another challenge is that TPE is non-selective, and there's maybe where we get into the immunoabsorption. It removes both the harmful substances, but also the beneficial plasma components, which gives less coagulation, and you have albumin depletion. Should we move forward to more selective apheresis techniques? Well, I think we just heard a brilliant talk about it. But for instance, in the Netherlands, the reimbursement is a real problem for the immunoabsorption. And it takes longer to do the treatment. So it's not that easy to implement these therapies. I think we also heard a lot about complications in the presentation by Bingel this morning. Fortunately, we're improving, because at first there were quite a lot of really big complications, and nowadays, I think it's a really safe therapy. We know what the biggest complications are, maybe hypotension, the hypocalcemia by the citrate, but you can manage that by giving calcium really early on. There's a little bit higher infection risk, And I think one of the still biggest challenges is the vascular access. And also that is improving with ultrasound-guided access, but I think we'll hear about it more in the coming days. And there's a nice overview, which is already a little bit older, about the adverse events, and I think it's a really safe therapy. But can we get any safer? There are also logistical and operational challenges if you think about therapeutic plasma exchange. It requires really highly trained staff, and as I said, in the Netherlands, in my center, we have 5 nurses that are really well trained, but training a nurse takes, well, we say about a year. And it's a small team, and if you need the nurse during the night and the weekend, it has impact on the schedule for the coming days. And there are already new procedures scheduled to collect all those CAR-Ts. And I think there's a lot of variation across centers, because the one center is much more experienced and does a lot more treatment than another center, but also between countries. And yeah, you could think, should we try to centralize all these treatments or not? But that gives a burden on the patient, because then patients need to travel quite far. And then there's the question of the replacement fluids, and it's been mentioned before today. I think it's always the question between albumin versus plasm, but as I said, each replacement fluid has its own trade-offs, but both of them are medication or are products that come from plasma from healthy donors, and there's a shortage. In the Netherlands, we're not self-sufficient. I think we've heard this before. We also still import part of the plasma. We're working really hard. Our colleagues from Sanquin, the Dutch blood supply, are working really hard to be self-sufficient. But if the indications are becoming more and more for the use of IVIG, then it still stays an issue. And it's a costly therapy, because IVIG is costly, but also the albumin and the plasma is a costly part of the therapy. So it's not only costly because of the replacement fluids, but it's also because of the equipment. If you need an apheresis machine, it's quite expensive. You need the staff, and we know really little about the cost effectiveness of these treatments. There has been done some work, but it's difficult. And as I said before, even in the Netherlands, I think that's a wealthy country. It's still difficult to find reimbursements for some of the apheresis technologies and therapies we would be able to give. And there's the practice variability. Differences in how much do you exchange, in the volume, the frequency, the duration. There's a lack of standardization across institutions, but maybe even within institutions. If one physician starts the treatment, maybe the advice is a bit different than if somebody else would be on call. I think there are also scientific and technological challenges. One thing that's really difficult, there's a lack of biomarkers, because of course you can measure some of the nice antibodies that has been named in the previous slides, but it takes time, and you want to follow up on those markers on your decision to keep going with the treatment or even finish the treatment. And what's the optimal duration of the treatment? What we not really know is who of our patients truly benefit from our therapy. It would be nice to know if a patient is going to really benefit or not, because then maybe we shouldn't start therapy. Of course, there are emerging technologies, and we heard about AI, but of course, the selective apheresis techniques are still emerging. They're, for some indications, really established, but for other indications, it may be less clear. And maybe in the future, and I don't know if it's going to happen, but maybe a robot is going to think about the decisions we just made. Are we going to start? Are we going to stop? I think it has a lot of potential, but how are we going to use it? And there are ethical and future challenges. I think we should realize that access to care is not normal for everybody. There are parts in the world that some care isn't available. So it gives disparities across Europe or across Europe and other parts of the world. And in lower resource settings, there's also often a limited availability of these therapies. And then there are the guidelines versus the real world practice. A very nice study has been done where they had taken a look at what says the guideline and what is done, and it really differs. And it's difficult because even though we have guidelines, most indications are not that clear in the guidelines or aren't in the guidelines at all. And then you should make a decision whether or not is apheresis, therapeutic plasma exchange is a useful part of treatment. And then there's the future, and some has been said about it by Miguel. He was quite uplifted about it. He thought, well, not a problem. But there may be potential competition from monoclonal antibodies and targeted immunotherapies. To my experience, because we see a lot of myasthenia gravis patients in our center, and there are a lot of new therapies, and they all sound wonderful, but all the patients end up in our ward again because they're failing or something happens. They switch from IV to subcutaneous, and then it's working less, and they have a relapse, and then they call us to start it up again. So I think maybe we won't be out of a job yet. But I think that's a question. Will therapeutic plasma exchange remain essential, or are we going to expire? What would I like for you to take home? I think it's very powerful. And in some patients, it is really, really a good therapy, but it's non-specific. The evidence is often limited or heterogeneously, and I think there's a real need for better standardization, reliable biomarkers, and more selective therapies. And that's where I would like to conclude. Thank you.
Thank you. Thank you, Claudia. I give you the word, Volker. We have time for questions. But I would like to stress one thing. Guidelines means rule of the thumb. It's helping us, and not more than helping us, in a way of thinking, in removing something from the bloodstream, which is giving a symptom complex. We don't cure in any way. So guidelines are more of a help for us and not a regulation or a standard in what being done, and that should be much more clear in the world. I see in many low and middle income countries, they read the Journal of Clinical Apheresis. I'm very happy with this journal, and you will also hear on Friday, but I think we should be aware that this is a rule of the thumb, and I give the word to Volker, who is one of the authors since 2012, of the co-authors of 2012 of the Journal of Clinical Apheresis special issue.
Well, thank you very much for these questions, and it's clear that I cannot agree with all things you said. I think first of all, guidelines is every time the past. It's not the future. It's what happened so far. And I think guidelines are very powerful to say that's our standing. And what you have said is correct and a little bit incorrect. It's not that unstandardized, because if you read the fifth section in every fact sheet, there is in 72% a concrete dose and schedule, what is in the literature, and even also the discussion for that. The second thing is, it's not that the apheresis doctor is in charge of a treatment schedule. It's the team. It's a multidisciplinary team. For example, we have treated ECP in children with different techniques because it's otherwise not feasible. But we combined it with immunoabsorption or with TPE in special cases, and also lung rejection. And I think the starting point is every time to know what to eliminate. Without a goal, you should not use a machine. And so I would say, yes, we have many goals, and we have, when it's clear and strategic, logical, that I can eliminate some of the physiological agents, then I should do it. And Esfh so far, and in combination with our Europeans, we have also the group TAOS, which is the treatment, the Therapeutic Apheresis Outcome Committee. And it's very hard to make these outcome rules, not why apheresis guidelines are so bad. The point is that medicine is that bad. Go to a hematologist, go to a nephrologist, go to a neurologist. Neurology is much more better, to say the truth. They have many scores. But go to a hematologist and ask him for a leukemia. When to stop what? If he would not have an international protocol, he could not answer that. So I think this makes clear that in our medical world there is much to do, and we should have in mind that we need an interdisciplinary team. The combination of TPE and other treatments, like Miguel said in TTP. I think it's clear today that what Gay Rock published many years ago, that we should treat it with plasma exchange, is not anymore true. Yeah, we should do it in a multidisciplinary surrounding. And I think that's what we should go in the future.
Yeah, I totally agree. And I think one of the important things, if you see the indication, so you have a goal and a plan, that you re-evaluate, so that you decide with the team when are we going to evaluate whether or not the treatment of TPE we start, the supportive treatment of TPE we started is effective, and do we go on or do we stop? But I totally agree with you, definitely. Yeah.
Any other questions? Oh, here.
Changing the rules
we have biomarkers. We have biomarker which is available in Czech Republic in 2 hours. I don't know, at the day, working day. The problem is weekend and night, but we have biomarker. I think TTP is a little bit different, but I liked your lecture. It was okay. But, yeah.
Thank you.
Sorry. Sorry.
Jsem na to myslela.
I think the most important thing is biomarker is not all thing. So, and we should not discuss good countries and not so good countries. But it's I remember 1996 when I was in Florida at Esfh meeting. I asked the colleagues why they don't measure CD34 positive cells before they make a stem cell apheresis, and they laughed and said it's so costly. Yeah. Yeah, but today it's routine. So we have it in 40 minutes. Other people have it in 2 days. So, and I think the point is we have to go back to the first question. When I take something away, which means apheresis in Greek, we should claim what it is. And then we have to look, is it okay or not okay for the patient? Because in 25% of all procedures, we do something new. And so there are many publications. Meanwhile, it's 12, so in the last 3 years, they looked in the rest of the plasma exchange. And so they could make the diagnosis. Even in immune absorption, they illustrated the antibodies, and so they found the responsible antibodies. So there are many questions, and biomarkers, I think, biomarkers is a good example. There is a devastating disease called GVHD. And when I'm correct, Nina, you may correct me, I think we are 32 years on the way to find biomarkers. And look how many we have. It's not so much. Yeah? And the belief in biomarkers instead of the logic of our therapy, I think that should be a good point. Yeah, no, it's okay.
I would like to end this session with a remark. 2,500 years ago, a well-known guy, Hippocrates, said already that disease reflects something in the blood, and you should do bloodletting. So, and he even didn't know how the blood was composed. So we still don't know a lot of things from what we are doing or what is happening in the blood and what we're taking away. Anyway, we see the result with that. And with that result, I'd like to end this session. Thank you again, and thank you also Kurt Derfler and Amlov. And I'd like to see you tomorrow again. This session will be ending now. Now we continue here, I think, in this ward with the General Assembly. So members of ESFH are invited to come to the General Assembly, and for the remainder at 6 o'clock, we find you back outside for a little
whatever circulates in your blood. If you're an ESFH member, please stay. If you're not a member yet, please become a member. Yeah. And otherwise, join us outside later.